Okay, welcome everyone. I am Gregory Fay. I'm a senior S researcher at the Institute of Mathematics in Toulouse in France. So on the behalf of the organizing committee, I am very pleased and enthusiastic about welcoming everyone to the 7th International Conference on Mathematical Neuroscience. So the first ICMNS conference was launched in 2015 by Olivier Fauvras in Juan-les-Pins back then, when we could still travel. And it has been organized 6 times since then. So due to the pandemic situation, this year, the conference will be held online, and that was also the case last year. So the aim of this conference, of this series of conference, is to bring together mathematicians and theoretical neuroscientists in order to use or develop or developed mathematical techniques for neuroscience problems. So this year, we will feature 3 plenary talks, and in a minute or two, we will start with our first plenary speakers, but we will also have 9 contributed mini symposia and 3 sessions of micro talks. So today, it's our great pleasure to start with this conference with Krešimir Jozic. So Krešimir is a professor in the Department of Mathematics at the University of Houston, and is also holding adjunct appointments in biology and chemistry, still at the University of Houston, but also in biosciences at Rice University. Krešimir holds his PhD in mathematics from Penn State University, and he got it in 1999. So since then, he has published more than 100 papers. He was awarded lots of several grants and also prizes. And his research topics include, but it's not inclusive, dynamical system theory, mathematical neuroscience, and I would say more broadly, how to use dynamical system, probability theory, statistics in mathematical biology. So today he has decided to give a talk on synaptic plasticity in correlated balanced network. And just before Krešimir starts, I just want to say that if people want to ask questions, they can ask questions at any time in the chat. And with Daniele, we will collect them and pass them to Krešimir at the end of his talk. Okay, so Krešimir, please, the room is for you.
Thanks a lot. So thanks for the invitation to this conference. It's been one of my favorites in the past few years when I attended, and I hope that we'll get a chance to see each other in person in the future again soon. So I'll talk about this question of synaptic plasticity and correlated balanced network. This is work with Alan Ackel, a graduate student at the University of Houston, Robert Rosenbaum, who is a professor at Notre Dame, and 2 experimental collaborators, Ariana Andre and Valentin Dragoy at the University of Texas in Houston Medical Center. So let me start just with a brief overview of the theory of balanced networks, and so where this kind of comes from. Again, this is not going to be complete. This would require a series of lectures to really explain in detail, but just to give you a highlight of what motivates what I'm going to talk about. So this goes back to about the early 90s, probably before then, when people noticed that the activity of in cortical cells is really irregular. So if you record from them, you see that the Poisson spike trains are really close to having Poisson statistics. Their CVs are around 1, maybe between 0.5 and 1. But if you create a model and you try to explain that, you take, for instance, an integrated fire model, a biophysically realistic model, and you assume that it takes more than 1 input spike to trigger a response. So n here is the number of input spikes necessary to trigger a response. So if you go to n is equal to 4 and n is equal to 51, for instance, you find that the coefficient of variation in the output of the model cells becomes much smaller than what you observe in experimental data, right? And that led to this question. So cortical cells should fire regularly, but do not. So why is that? Right. So here they conclude, Sofsky and Kaw here conclude that neither the data nor the models are really controversial. These are pretty standards, but they're not consistent. So what's going on? And so there's couple possibilities, one strong contender that emerged, I think, a few years later, is in this paper von Briesig and Polinsky that I'm sure most of you know. This is the one that introduced in '96 the concept of a balanced network is a self-balanced network. The model is a relatively simple one, relatively widely accepted one. You have a subpopulation of excitatory cells, subpopulation of inhibitory cell, and a strong external input driving the activity of both. And you see that the network self-balances. The important thing here is that the synapse strength scales like 1 over square root of n, where n is the number of cells in the network. I'll get back to that point in a little bit later on. And what happens is that the excitatory inputs, this is, I hope you see my mouse here, the excitatory inputs, these are the ones above here, and the inhibitory inputs, these one below here, approximately cancel so that the input currents to the individual cells in the network are approximately 0, and the spikes are driven by occasional crossings of the excursion from this 0 baseline, resulting in a relatively irregular spike trains of close to CV is equal to 1. So these networks are not only, this is not only a computational theory, this is actually, there's a nice mean field theory, which I'll give an overview of in a moment, that accompanies this particular theory as well. So, so that was one of the solutions. So since then, the theory of balanced networks has become really important in theoretical neuroscience. There's a bunch of, this is just a small sample of all the papers that you will find if you look into what followed the original publication von Briesig and Polinsky. So there's a bunch of theoretical papers that I put here on the left, and also a bunch of experimental papers, 3 of which I've selected here on the right, that support the idea that some sort of balance does occur in cortical networks, in cortical circuits. And so importantly, as I mentioned before, is this 1 over square root of n scaling that of the synapse strengths. And here's a paper by Barral and Reyes that points out that may in fact, there's some experimental evidence for that particular scaling in organotypic slices, sorry, organotypic cultures. The important thing here is that there is some support for the theory, for this theory of balance. Again, what exact balance occurs is still somewhat debated. Is the balance tight? Is it sort of more sloppy? That's still not completely resolved, but I think it's fairly well accepted that networks, cortical networks, are in a balanced state. Okay, so let me also mention just briefly, in the original theory, the networks are in uncorrelated states, right? So you have the excitatory input to the network, the feed-forward drive to the networks is typically uncorrelated in the original theory, and that results in the recurrent network, this network of E and I cells that I've shown you before, going into an uncorrelated state. In fact, the correlations drop to 0 as the network size increased, right? And now that's not really something that you see, so this would be the corresponding activity. This is not really something you see in cortical networks. Many cells could be weakly correlated, but there's certainly pairs of cells, and I think this is not disputed any longer. There are at least some pairs of cells that can be moderately or strongly correlated. And the question is, how do you create a theory that also produces a balanced network theory that also produces a correlated state? And the way to do that is to actually include some correlations, some structure in the network connectivity, for instance. Here in this paper right here, Rosenbaum et al. Looked at, Robert Rosenbaum et al., they looked at a spatially extended network, spatially structured networks where the footprint of excitation and inhibition are incommensurate, and that can lead to correlated states. And also, if you assume that the feed-forward input to the recurrent EI network is correlated itself, then that can result in correlations in the recurrent network, in the spiking of the E and I cells in the recurrent network, that in the balanced state as well. And so that's the typical activity that you see in those cases. The level of correlations can be governed, and I'll view this in a moment, by the level of correlations in the feed-forward input. So the correlations in the feed-forward input govern the amount of correlations or the level of correlations in the recurrent input, control it. So, and this is, there's other solutions as well that I'm not mentioning right here to this puzzle of correlations. But I'll take this paper, this solution is proposed in a paper by Baker et al. From Robert Rosenbaum's lab as the one that we use to model correlated networks, correlated states. Now, as I said in the beginning, we're going to talk about plasticity in correlated balanced networks. So I don't think I have to convince you about the importance of plasticity in neuroscience. This is how we think cortical networks or model networks learn, not just cortical networks, just networks in general across species, even species that don't have cortices, change and adapt to learn new behaviors, responses to new stimuli, and so on. So it would be impossible for me to really give you a complete, even a partial review of the theory of plasticity. And one thing that's maybe sometimes neglected when we talked about plasticity is the fact that plasticity also has probably a homeostatic role in cortical networks, or again in neural networks, right? Unlike, the emphasis typically in machine learning is not about the dynamics of these, let's say, feed-forward neural networks in a machine learning applications. The dynamics is kind of less interest. It's the changes in synaptic weights and the responses that we see to particular inputs that are of interest. But in biological networks, whether the network itself is close to a dynamical state, close to an operating point that's defined, let's say, by the rates being in a particular range and the correlations being in a particular rates, that may also be important. And the plasticity may actually, different plasticity mechanisms may be there to guide the activity, to keep the activity of the network within those operational ranges. And this is something I'll get back to towards the end of this lecture. In particular, the types of plasticity I'm going to talk about here are spike time dependent plasticity. Here are a few examples. I'm sure you're familiar with the first picture. This is the typical Hebbian example. If a presynaptic spike and a spike in a presynaptic cell precedes a spike in a postsynaptic cell, what you get typically is long-term potentiation of a synapse. If a spike in a presynaptic cell comes after a spike in a postsynaptic cell, you get into this red region and the synapse gets depressed. Right, there's different examples, experimentally observed examples of this types of plasticity, and you also see here in electric lateral line in the electric fish. Forget what EL is. I think the lateral line in the electric fish. You also see an opposite, an anti-Hebbian type of plasticity, where exactly the opposite happens. The before post results in a depression of that synapse. And so there's many other types of plasticity as well that I've cut these from this review paper by Abbott and Nelson, and there's a few that I've cut out, and you can also find other ones in the literature, many other ones in the literature as well that have different shapes and different effects. The question that we're going to ask here is, can we extend the mean field theory of balanced networks to include plasticity, right? So this is something that we actually, even though the theory of balanced network is well developed, I don't think we've looked at the literature, and this is not something we've seen answered. And importantly, so just extending the theory doesn't really matter, but what is important is to, I think, understand under what conditions can the plasticity, can changes in the synapses in the network preserve the dynamical state of the network, under which condition, on what types of plasticity are going to keep the network in a balanced state, in this irregular spiking state, maybe correlated state, right? And so that's the question that we started with. And the answer is yes. What you can find is, if you assume that there's a separation of timescale, if you assume that synapses change relatively slow compared to the timescale of the rates, the interspike intervals, then you can in fact find a closed set of equations that describe the evolution of the synaptic weights, of the mean synaptic weights in the network, right? And as a result, you can actually also compute how the resulting rates change in the network of the different subpopulations, and how the covariances change, or correlations change in the network as well, along with the rates and the weights. All right, and so let me just give you a little bit of background. What we use here is the network model is, again, a recurrent network consisting of excitatory and inhibitory cells. with a external population providing feed-forward input to the recurrent network. The network, again, you need a strong feed-forward input to put the network into a balanced state. The network itself consists of exponential integrating fire neurons. The particular model here does not really matter. We could use leaky integrating fire neurons, or any of your favorite, even some biophysically realistic models. The important thing is that we did use current-based synapses rather than voltage-based synapses. The theory can probably be extended to voltage-based synapses, although there's some changes in the scaling that we'd have to consider. And again, the input currents here consists of two parts. There's a recurrent part, RJA, and a feed-forward external input, XJ, to each cell to drive the activity of each cell. Again, the superscript here, A and B, are going to denote the type of cell we're looking at, excitatory, inhibitory, subpopulation, and the subscript J here is going to refer to the cell number. We're typically often going to do the mean field theory. We're going to average over all the cells in a network to find the average activity in the excitatory and the average activity in the inhibitory population. The synaptic weights are given by this block type matrix. It consists of EE weights, EI weights, I, and so on. The unscaled weights are given by a lowercase JAB. So this is just the weight between the two population types. For instance, at the beginning, this is how we initiate the network. And the important thing is that we multiply the weights by 1 over square root of n. So where n is, again, is the population size, right? The weights are scaled by the square root of the population size. Again, as I said before, this is important for achieving the balance state, for putting the network in this regular spiking balanced state. In the mean field theory, what we do is we average all the weights across the entire network, right? So you take each pair of subpopulations, E, I, and so on, IE. So again, the second, as usual, the second letter in a subscript denotes the presynaptic population. The first letter denotes the postsynaptic population, right? So this would be the average weight of a synapse between the inhibitory and an excitatory neuron, right? And the way you can average this by multiplying the probability of a connection between a cell in the population B to a cell in population A. Here is the weight of that synaptic connection. This is the unscaled weight. We did not multiply by 1 over square root of n here yet. And QB is the proportion of cells in the network of that particular type. So for instance, QE would be 0.8 if you have 80% excitatory cells. Similarly, you can compute the average feed-forward connectivity matrix, right? So there's the average strength of connections from the external population to each type of cell, each population in the network. And the average rates would be obtained by averaging the rates within the excitatory and within the inhibitory population. Okay, so those are the three quantities that you typically, those are the three quantities that you get by averaging across the entire populations. Subpopulations that constitute the recurrent network, and you can compute the input to each cell type, the excitatory input and the excitatory cells and the inhibitory cells, by looking at the average recurrent input, right, which is the average weight matrix multiplied by the rates of the inhibitory and excitatory weights. That's the vector R here, plus the average connectivity matrix, from the external input, from feed-forward input, times the rates, the average rates in the feed-forward population, right? In the population, external population that's driving the activity in the network. Now, remember, each one of these weights is scaled by 1 over square root of n. There's n of them, so n divided by 1 over square root of n gives you a square root of n factor in the actual current that's driving the network. So that, of course, would blow up as the network size increases, unless this term in the parentheses here is 0, right? And so the balance state, the surprising thing about the balance state is that, in fact, this cancellation of these two terms defines a stable state of the network, can be under certain conditions, can define a network state that's actually stable. And in that state, the rate is exactly given by solving for R, right? The rate vector is given by solving for R when setting the term in the parentheses in this average input to each cell type equal to 0, right? And so this gives you an expression, an exact expression that works in the limit as n goes to infinity of what the rates in the network and the average rate in the excitatory and inhibitory subpopulations in the network should be, right? And again, in this case, the firing is going to be uncorrelated, become uncorrelated as the size of the network goes to infinity. Now, to get a correlated balance state, what we do, again, this is not the only solution, but it's the one we chose for this particular case, is to assume that the input cells, right, the cells in the external populations that drive, again, the activity in the recurrent network, the inputs here are correlated themselves. Under those conditions, I'm just going to give you the result here. Under those conditions, the correlations do not go to 0. They remain order 1 as the network size grows. And in fact, you can find a closed form expressions for the covariances between two cells, for the activity of two cells. You take the activity window of size t window, you count the number of spikes, that's a random variable, and the average covariances between two cells of different types, like the E cell and EI cell and so on, and between an I cell and an E cell and so on, is given by the expression that I've given you down here. Again, this is an expression that depends on the covariance, on the correlation between cells in the input, the activity in the input, and the average matrices, the average weight matrices that are defined on the previous slide. Again, this is a quantity that you can compute exactly in this particular term. This is again following the theory of Cody Baker and from Robert and others, from a paper that came from Robert Rosenbaum's lab, who's Robert again is a collaborator on this particular project. Plasticity. Okay, so how do we model plasticity in this network? What we do is a relatively standard approach this is given in terms of eligibility traces, right? So an eligibility traces is simply a filtering of a input spike train. So every time you have a spike train SJI in a cell J in the population A, every time there is a spike in the cell J, the eligibility trace, it gets kicked up by one unit, right? And then it decays back down at a rate that's given by tau STDP. That's a time scale that determines the time scale of this plasticity window, really. It decays back, the eligibility trace decays back to the baseline level at that particular time scale. Okay, another spike, another kick, followed by a decay. See the last spike over here gives the last kick, and then the activity decays back almost to baseline before another spike kicks it back up, right? So it's just a simple filtering, right? So the way this then works, how we get a plasticity rule out of this using the eligibility traces can be demonstrated using classical Hebbian plasticity. So in this particular case, let's say we have a spike in a presynaptic cell, right? So we have a synapse from cell K to cell J. Let's say both of them are in the excitatory population, just to keep things simple. This is not necessarily important for this example, but suppose we have a spike in the presynaptic cell. As a result of that, the eligibility trace goes up and decays back to 0 at a particular rate. If this is then followed by a spike in the postsynaptic cell, SJE, within a particular before the eligibility trace has decayed to 0, we would expect the weight, the synaptic weight J sub JK to be potentiated, right? So we can do is take the product of the SJE and the eligibility trace, right? If that's large enough, if that's non-zero, we would increase the weight from the presynaptic to the postsynaptic cell, right? And so this is the term that corresponds to that increase in the weight. Of course, that increase is going to be small. The larger the distance, the larger the time between the pre and the postsynaptic spike, the smaller the effect of this pairing. And the effect can also be scaled by some constant that you choose that determines the size of the effect of the pairing as well. This is something that's controlled in the model or would be maybe determined experimentally. Again, if the postsynaptic spike comes before the presynaptic spike, right? So this is post followed by pre. The Hebbian rule would predict that that's going to result in the weakening of that particular synapse. Again, you can get that by multiplying now the eligibility trace in the postsynaptic cell by the activity in the presynaptic cell. And if that is non-zero, you now have a negative contribution to the rate of change of the synaptic weight. And that is exactly what you have in this particular model. This whole thing can then be multiplied by eta E. The reason I'm multiplying by this term eta is to emphasize that we will need many spikes. We assume that we need many spike pairings to get a non-negligible effect on the synapse weight, right? So the synapse changes are relatively slow. And that's exactly, if you combine all this together, you exactly get the classical picture of Hebbian plasticity, a spike time dependent Hebbian plasticity. Pre-post goes up, post-pre results in a decrease in the synaptic weight. Okay, you can get this, the general idea right here is captured by this equation right here that essentially captures a lot of different synaptic rules. So this single equation right here describes a whole bunch of synaptic rules. You have a scale, the time scale, 1 over the time scale is this term eta, which we assume to be small. There's a drift term, A naught. There's a rate dependent term right here. This is a term that depends only on the activity of the presynaptic or the postsynaptic cell. And here is a pairing term, right? You pair a eligibility trace in either the presynaptic or a postsynaptic cell with the activity with the spike train, again, in either a presynaptic or a postsynaptic cell, right? And so if you combine these in different ways, use different coefficients, right here, you can get a whole slew of different plasticity rules. For instance, classical, you don't have to, I'm not going to go through these in detail. I just want to list them as particular examples of this general type of plasticity rule. You get classical Hebbian plasticity, anti-Hebbian plasticity, homeostatic inhibitory, which I'll get back to in a moment. Oyer's rule, which you probably heard of, Cohonen's rule, which again is something we're going to use in the examples that follow, right? So the main point here is that you can capture a whole bunch of different rules with this general expression right here. What you can't capture in this expression is triplet rules, which would involve the next term in this particular expansion, if you wish. Now, the fact that eta is small allows us to average in time. And since we're looking at a mean field theory, we can also average over different network realizations. Really, what we average is over all the J's and K's in the population A and the population B. Right, and as a result, if you do this, again, I'm skipping a bunch of details here in the interest of time. I'll give you the citation at the end of the talk. You get an average equations that give you the average evolution of the weights, right, in terms of the rates of the populations and the covariances in the population. Excuse me. And the rate term, again, depends only on the rates, has a term that depends only on the rate of the presynaptic or a postsynaptic cell, and it has another term that depends on the product of the rate in the presynaptic and the postsynaptic cell. And the covariance term is given in terms of the Fourier transform of the synaptic kernel, as well as the Fourier transform of the average of the spike trains. Right. So this is what you typically see. Again, this is a little bit easier to compute. And that's why we use the Fourier transform of these terms rather than looking at it in the time domain. This is just the spectral density of the spectral cross-spectral density of the two spike trains. Now, how do we close the equations using this theory? Okay, so we have an equation for the firing rates. We have a closed equation. We have an equation for the covariances, right? And we have an equation obtained by averaging for the synaptic weights, or the mean field equation for the synaptic weights, which depends only on the rates and the covariances, right? So the important thing is that the first two equations are fast, so we can assume that they equilibrate relatively quickly, and the only thing that changes are the synaptic weights, right? And if you look here on the right-hand side of these equations, of the equations for the rates, this equation really depends only. The only thing that changes in these equations, variable, that's not a parameter, is really the weights between, the average weights between the different populations, right? A and B, let's say E and I and I, and so on. This is the only thing that varies in the equation W bar. Wx is fixed. That's the synaptic wave from for the external input. And similarly, the same thing is true here. I've rewritten this equation a little bit more simply, but the only thing that determines the covariance is, again, are the average synaptic weights between the two populations. So you can solve for the rates as a function of only the synaptic weights. You can solve for the covariance as a function of only the synaptic weights. You can plug these into the right-hand side, and it gives you a closed form equation for the weights themselves, right? So you now close them, you removed R, you move the covariances and the rates from the equation, you get the equation, self-consistent equation or a closed equation for the synaptic weights. If you want, if now those weights converge to a stable fixed point, right? If the equation, now this would be a one-dimensional equation, if only one type of synapses change, if they converge to a stable fixed point, you can find that stable fixed point. Right? That would be, say, JAB star is the value of the stable fixed point. You can plug back into the first two equations, determine the rates and determine the covariances, and in fact find the predicted rates and the covariances in that particular state. So just to test the theory, what we've done is look at, let me go through this again because I went back. If you want to look at to see what the predictions of these theory hold, what we looked at is a particular type of rule. It's called Cohen's rule. It's just a simple one. It's a type of anti-Hebbian rule, which has a term. The first term here is essentially an activity-dependent term, which is going to decrease the synaptic weights of individual synapses, but dependent on the activity of the postsynaptic cell. And if every time there is a post before pre-pairing, right? It's anti-Hebbian, post before pre, the synapse is going to be potentiated, right? And so this is, if you look back, this is a rule that's been proposed in artificial neural networks, and there's some arguments about whether it's biologically realistic or not. Now, we use it more as an example to kind of test whether the theory is correct. And in fact, what we find is that as the network size grows, the theory predicts, right? You find again we find that the network the theory predicts that the network stays in a balanced state. There's no blow up of the rates. The weights should approach a stable fixed point, which is given by the dashed line over here, right? And in fact, as the network size go, the solid line here is showing the computational simulations, the simulated balanced network of the type of I've described before. The simulations effectively agree pretty well with the predictions of the theory. Again, the predictions get better and better. This is an asymptotic theory that works in the limit of a large network. Of course, the theory agrees with the numerical experiments better for larger network sizes. Similarly, the rates that are predicted by the theory also agree with the simulations, and the theory and the simulations also show that the covariances decay and the correlations in the network go to zero as the network size increases. And the correlated state, so this is again, now we look at this was the asynchronous uncorrelated state. The input to the network was a CX was 0. In the second case, we assume that CX is non-zero. You get a similarly good agreement between the two. The main difference right here is that the covariances do not decay to 0. They stay order 1. Right, and so here's different examples where the different EE, EI, and II covariances are different because of the different solutions, the different structure of the network, the average weights and the recurrence weights. And so one thing that the Cohen's rule also does is the following. So if you look here, if you look at the first term, this term is this describes this anti-Hebbian nature of the plasticity rule, and it says that if there's a pairing, a pre-pairing in the spikes of the two cells of the presynaptic and postsynaptic cell, then the synaptic weight gets potentiated, right? So if there's sufficiently many of those pairings, this particular rule will predict that the network, that the activity should run away, right? I mean, just intuitively, you will expect that that's going to happen, right? And especially if the pairing leads to a sufficiently strong potentiation of that particular synapse. And indeed, this is what you observe. In fact, if you look at the closed form, this closed solution of this particular rule, right? So this is, if you close the equation as I described before, you'll see that this, now you get a one-dimensional equation. You can study the bifurcations in terms of the bifurcation parameter beta that describes the strength of this interaction. And you see that as the larger beta is, the network essentially undergoes a saddle node bifurcation, and there's no longer a stable state for the synaptic weight to go to, right? You expect to blow up in the synaptic weights as a result, as beta becomes sufficiently strong. And since the synaptic weights blow up between excitatory cells, you expect the excitatory rates to also blow up. And in fact, this is what you see in the numerical experiments as well. The difference here is that this blow up in the numerical experiment, simulations, happens at a value of beta that's much lower than predicted by the theory. That's not necessarily unexpected, because we're ignoring all the heterogeneity in the network, all sources of noise in the network, and this is probably a noise-induced bifurcation. So this is effectively, if you're in a regime right here, where you're nearby a bifurcation, a small heterogeneity in the network, a small source of noise effectively in the network can push you across that stability threshold and result in an instability, right? So this is typical in, if you look at cell node bifurcations in the presence of noise. To get a better handle on this, we would have to include different noise terms, which would involve a extension of the theory to higher order terms and so on, which we haven't done. Correlations. Correlations actually have a relatively small impact. Let me just focus on the two right-hand pictures right here. If you change the input correlations, in the case of the Cohonen's rule, the excitatory weights in equilibrium change relatively little. The excitatory rates and inhibitory rates in equilibrium also change relatively little, right? So the input correlations do not affect the stable state in the network, and this is something that's been observed before. For example, by Graupner and all this, there's a number of paper. This happens unless you really fine-tune the learning rule, right? If you have a very particular types of learning rules, for very particular types of learning rules, the correlations can matter, but it again has to be adjusted just right. Okay, let me actually go forward and to experimental data, because I think that's going to be to have some time to go over this and talk about the paradox. Talk about essentially how we applied some of this theory to experimental data, and to kind of motivate this. Let me start with this. This is a really, I think, important paper recent paper by Zenke, Gerstner, and Ganguly. It's a 2020 paper that argues that typical types of homeostatic plasticity that we now use in models are much too slow to keep Hebbian plasticity from running away and causing a runaway effect, essentially blow up in networks, right? So there must be some types of rapid compensatory processes that would prevent it, that do not occur on the time scale of hours or days, but occur on much shorter time scale to keep the network close to the operating points that we know they are in. Right, and so what those are is a question, right? And so in the next few slides, I'm going to suggest that that a particular type of inhibitor plasticity may actually play that role. So the experiment, this is again experiment done by Arena Andre in the lab of Valentin Dragoy at the University of Texas Medical Center in Houston. It's a very simple experiment. So the idea is that you inject a certain virus in V1 to allow for optogenetic stimulation of excitatory cells and subnetworks of V1. Circuits of V1. So these are the pyramidal cells are being excited. You can excite in these cells by using light. What we then predict, it is associated plasticity would increase network correlations, for instance, or network rates as well, and the compensatory processes could then affect the correlations and the rates in those networks, right? And the experiment, the behavioral part of the experiment is really simple. You have a monkey, it fixates. There's stimulation, laser stimulation, right? There's no other type of input. The monkey does just fix it at this particular point, and then there's a response at the end. It has to move sides left or right, right? But this is really the time before the response is the one that we're going to be interested in, where you stimulate the network. Okay, the firing rate reliably go up during stimulation, right? So you record from this population, you look at the average. So these blocks are the trials. Each trial is of a few seconds in length. Each block trial, like they're separated into blocks, and the blocks are a few minutes in length. And then you have a session which consists of 40 blocks. And what happens is that the firing rate stays roughly constant across the entire section. Every time you stimulate, you increase the firing rates of the cells that are being stimulated. Presumably recurrently connected, and therefore all cells increase their firing, right? Even the unstimulated cells are also being pulled up or fire more as a result of stimulation. But what happens with the correlations? If you compute the correlations between individual cells in these populations, the correlations start positive at the beginning, right, and the first few blocks, and then drop close to zero across the blocks. Right, and this is kind of what a typical example shows, right? So this is an example session of monkey one. So the two monkeys that were trained to do this. The difference between laser stimulation and control. First, during laser stimulation, you have an increase in the correlations, and eventually across the trials that drops to zero. This is an example in monkey two. This is pretty consistent. But also what you find is that during rest, this does not happen, right? So this is again during awake. There's an example in awake. You high correlations at the beginning, during the laser on period, and again the decay to zero. And during rest, when the animal has its eyes closed, it doesn't perform any task, right? So it's just kind of resting. It's not unclear whether it's sleeping really. And during those times, these correlations in fact remain constant, right? So the network is presumably in a different state where the effect of laser stimulation does not affect correlations between individual cells in the populations in the same way as it does in the awake state. Okay, so what we did here, we used this example of homeostatic plasticity. Homeostatic plasticity is a type of plasticity. It's introduced by I had a couple of slides before, but I skipped forward an inch of time. This is a type of plasticity that's introduced, that's really studied. We used inspiration from a science paper by Vogels et al. And the idea of this homeostatic plasticity, really in those original papers, is that it keeps the firing rates of excitatory cells in check by regulating the synaptic strength onto the excitatory cells from inhibitors. So the I to E connections are either potentiated or decreased to keep the firing rates of excitatory cells in check. This is a type of plasticity that falls well into, it's exactly of the type that we can study using the theory that I've explained before. Right, and here's the type, here's a typical picture that it's a rate dependent picture right here, so it's not static right here, but this really gives you an idea if there's a pairing between pre and post sufficiently close, the synapse is potentiated. It's potentiated in a rate dependent way so as to keep the excitatory firing rates at within a particular range. What you find also is that this particular plasticity rule is also going to decorrelate the response of the stimulated cells in a computational in a model network in the same way that as observed in the experiments, right? So after this is you have to tune the tau, the time scale of the plasticity. That's the only thing you really have to tune here really carefully to make it sufficiently fast, right? If you make it too slow, if you make it act over hours, you're not going to see what we observe here. But if it's sufficiently fast, what you see is exactly the type of decay of correlations, the decrease in correlations across 40 trial blocks, the same time scale as observed in the experiments that we observed in the experimental data. All right. It also shows a relatively small effect. You can know that the way we assume that the network changes during a resting period is by downscaling the synapses. And there's some support, experimental support for this in the literature. We downscale all the synapses in the network, and we do use the same protocol as an experiment again, and you find that the change in the correlations in the resting state is going to be much smaller than in the awake state, right? So here are the two states. I'll just concentrate on this bottom figure right here. Right, so in the resting state, in the resting network, the change in the correlations is there, but it's much smaller. So this is again consistent, at least not exactly, but fairly consistent with what we see experimentally. And so this provided a hypothesis, right? So one way that this homeostatic plasticity, one form of homeostatic plasticity that could push the network into this uncorrelated state under laser stimulation could be this type of inhibitory plasticity that I described in this example that we use in this network. And so what Ariana did, she went back, and there's a way to identify putative excitatory and putative inhibitory units in the network by looking at their spike shape. So narrow spiking units are the ones that are putative inhibitory cells, and broad spiking units are putative excitatory cells. And you can then compute is a cross correlation between cell pairs, right? Before, at the early stages of the stimulation, and then in late stages of the stimulation. And see, that is a proxy which you can use for a change in the interaction strengths between those cells, okay? And what you find is that the interaction between broad pairs, meaning putative excitatory cells, remains approximately constant across the experiments. So cells interactions, certain pairs have much stronger interactions after stimulation, right? So that is consistent theory that that it's this type of inhibitory to excitatory synapses that are being put relatively short time scales that result in the decrease in correlations that we observed experimentally. Okay, that's about it. So conclusions. So what I've described is a general framework to model the effects of STDP on network dynamics, predicts the state of the network and the average quantity, the mean field. It's a mean field theory. So it does involve higher order effects. It doesn't involve any noise and so on. So this would be an extension of the theory that I think would be interesting. The approach is flexible, can be extended for multiple inhibitory subtypes, multiple sorts of inputs, subpopulations of different types, and so on. It just increases the size of this interaction matrix. And I think you can even think about how to extend it to higher order STDP rules. So for instance, triplets and so on. All right, this is the next point right here. And what we haven't really thought about is to extend it to cases where the external input is not strong. So these would not be this tight sort of balance. If you're familiar with this paper by Hamadi and Miller, this paper by Hamadi and Miller, we are really in this regime where the external input is really strong. If you go to the state, to this perhaps more realistic state where the external input is not as strong, I think a related mean field theory can be developed, and I think the results would be relatively similar. And so here's the reference for the theoretical part. We're currently writing about the experimental part. Again, my collaborators right here, and I'd be happy to answer questions. Thank you.
Thank you. I already have 2 questions for you from the audience. So I will start reading the first one. So it's from Yuri Rodriguez. So actually it's a 2 question in one. So what is the physiological meaning of an eligibility trace? And is it to mimic an enzyme or a slow integrative process in the neuronal?
The eligibility trace is so it's really an abstract concept, right? So that captures as a reduction of a particular interactions that captures the particular effect of inhibition. And so the way this is done is the way plasticity works is really complicated, right? I mean, there's this calcium influx that stays into the synaptic into the spines, that is on for a certain time, that provides a window of opportunity for the synapse to be potentiated and so on, right? And so this does away with all this complicated biology and captures it into a really oversimplified oversimplified particular interaction. And so make a direct connection to biology is relatively hard, right? But it does capture the spirit of the biological processes, right? And it's pretty frequently used. What's the second type? But I wasn't sure about the enzyme question.
Yeah, so maybe because I have multiple other questions, I will switch. I mean, you already answered one question from Yuri, so maybe I can jump to Jonathan Rubin's question. So he's saying in the theory first presented here, input correlations have a strong impact on correlations within the network. So in the theory with plasticity, the results seem to show that input correlation have little effect. So how to these results, I mean, how to fit together these results?
So sorry, could you, so the input correlations have, could you repeat the question? I'm sorry.
Input correlations have a strong impact on correlation within the network. But when you were talking with the one within the plasticity framework, the results seem to show that input correlations have little effect.
Right. Okay. So there's a little bit of there's a difference right here, right? So what we've shown is that the correlations, the input from the external population, right? So the correlations in the external population have a small effect on the steady state of the network, right? What I haven't talked about is how the correlations between cells in the network change, right? That's a separate point. So the first point was that the input correlations from the input affect the steady state of the network relatively little. But what can happen is that the correlations between cells in the network can be affected by plasticity. Right? So those are two different things, right? So the steady state is not affected by input correlations, but the steady state correlations can be affected by a plasticity rule. And that's shown in the second example.
Okay. So I have a third question by Olivier Faugeras, who is asking, do correlations in the input result in correlations in the network rates and in the coupling coefficients, or in both?
Do correlation. So what happens is, so let me give you let me kind of answer the questions. Sometimes it's interesting to answer the questions. I think this goes to it. So the input correlation. So what frequently happens, and this is something that I think is really interesting. I skipped over it. Is that correlations, and not just the correlations, but actually just the input itself can produce correlations between weights and rates. Okay, so this is something that we usually ignore in balanced networks, correlations between weights and rates.
Okay.
And if you look at the theory, this is you just take the average, you assume, you use the usual trick, you assume that these are uncorrelated, and that gives you the nice theory that I described in the beginning. What frequently happens, plasticity rules can in fact introduce those correlations, and they essentially make the mean field theory, that the mean field theory can then break down. And to account for that, this is a little bit, it's in the paper here, discuss this briefly. To account for that, you would have to extend the theory to take into account correlations between weights and rates. And as far as I know, that has not completely worked out yet. But I think that's an interesting direction. I think this is something that will happen in plastic networks. I mean, those types of correlations between weights and rates, the fact that are to have the, and therefore the random weights are going to be correlated in the final state is something that will appear, and it's something that the balance theory, balance theory does not account for.
Okay, so I think that did we lose? No, okay, sorry. Maybe my internet connection is not that good. I guess that we have multiple questions. I'm not sure that we have time to take all of them, just because in 5 minutes we're starting a mini symposia. So I'm just taking one more, and then I'm sorry for the others. I guess that maybe you can people who have not asked their question, they can send you an email, and I'm sure that you will be more than pleased to answer them. So I will take the question about the 1 over square root n scaling. So is it preserved with by the ongoing plasticity, or is it just basically put there by the, you showed us the fixed point, and this gives you the kind of a rule so that you would have the scaling needed?
Yes, thanks. Yeah, no, you assume that is there, right? That is unchanged by the plasticity rule. The 1 over square root of n just stays outside in this theory, right? Yeah, that's I don't know what more to say about that. Yeah, but we can argue. This is again a mathematical convenience, and we can argue about how realistic that is and what support there is for those rules. But that's something that is all in all theories of there's extensions to conductance-based synapses that have a different scaling rule, but in the balance theory, that's something that needs to be there.
Okay.
Good question though. Yeah.
Thank you very much, Krishimir. So, well, I mean, I'm pretty sure that you will be seeing all the other questions that have been asked, but maybe you can answer to the people directly afterwards. So now you can, there is 2 mini symposia that will be run in parallel. So if you stay here, it will be chaired by Jonathan Rubin. Here I can see him with a huge soccer ball behind him. And it's going to be on dynamic ion concentrations and neuronal network dynamics. So this is channel 1. This is where we are. And on channel 2, this will be mean field models in neuroscience. And thanks again, Krishimir, and enjoy the rest of the conference. Bye.
Yeah, thanks, Gregory and Kresha. I think my mic is now on, so I'm gonna sort of stay put. I see Dave. Hello.
Hi, John. Can you hear me?
Yeah, you're good.
Okay.
We'll start your talk at 10. I don't think we have to do a whole lot. I think you can just. Do you see if you have permission to share your screen?
Yeah, I'm pretty sure I do.
All right.
So I'll wait.
Sure. I mean, do you want to try it? Do you want to see if your slides will show up? No, it looks good.
It worked?
Worked.
Wow. Let me see if I can get rid of
part of your desktop and part of your presentation. And I assume if you just go to full screen.
Yeah, I'll try.
Yeah, that little button there should. Yep. Okay. So you should be good. I hear you fine. I see you fine. Yep. So. Looks like we're still being recorded, so I guess we should keep that in mind. I thought I would.
We'll trim the recording, but certainly you're live on YouTube.
Okay, we're live. Okay.
That I cannot cancel.
Right. Well, that's okay. I'm not planning anything wild. I thought I would go with the soccer ball background to sort of acknowledge people who are skipping European Championship watching to attend this conference, which I know is a sacrifice. So I appreciate that, but hopefully it will be a good choice. I guess just as I'm with the time at hand, I can just start talking about the session so as to not take up speakers' time. So this session is about some of the various effects that impact neuronal firing that often get ignored or neglected in theory. right? So we have dynamics of things like ion concentrations, neuromodulators, blood flow, all of which, you know, the mean field theory is beautiful, but without these effects, it may be completely missing the boat on some important neuronal phenomena. So part of the point of this session is to call attention to some of those effects and see how people are dealing with them in the theory these days. So that's all I want to say about the session. I have 10 o'clock, so let me introduce our first speaker is David Terman from The Ohio State University, where I was fortunate enough to spend a few years. I learned a lot about giving talks from Dave and always enjoy his presentations. And now we'll hear the latest work he's doing on the dependence of neuronal firing on cerebral capillary blood flow. So Dave, the show is yours. And please just type questions in the chat. And we'll
let me know if there's anything in the chat, because often I miss those. But
clarification, I'll save it for the end.
Okay. Well, thanks, John, for inviting me. It's really a pleasure to do this. This is joint work with a surgeon on campus, Yosef Hassani, who works on issues related to stroke and small vessel disease, and an undergraduate, Liam Chen. And I'm going to talk about, in this short talk, I'm going to try to combine three models, actually. One for blood flow, one for the electrical activity of neurons, and one for ATP production by the mitochondria. So the idea is that, blood releases oxygen into the tissue, and the oxygen is used by the mitochondria to produce ATP. So that's the energy. The energy ATP is used to maintain the pumps, sodium, potassium pump. And of course, the pump is needed to maintain the ion gradients, and the ion gradients are needed to maintain spiking. So if you have impaired blood flow, the oxygen levels will go down, the ATP levels will go down, the pump strengths will go down, and then potassium will accumulate in the extracellular space, because every time the neuron fires, it releases potassium into the extracellular space. And if the extracellular potassium gets too high, that leads to bad things. Depolarization block, spreading depression, spreading depolarization, stroke, and things like that. So here is just some numerics. If the ATP level is high enough, then, sorry, I'm not very good at this. The cell can maintain spiking at some rate, and the potassium, let's see if I can do this. I think I had too much coffee today. Oh, well. The potassium levels off at some normal level, but if the ATP level is low, then as the cell spikes, the potassium builds up, and if it gets above some critical value, then the cell will go into depolarization block, and that could initiate spreading depression or depolarization. So here's some questions we were interested in. Is one, very basic: how do changes in blood flow impact a neuron's ability to maintain firing at a given frequency? So presumably, the faster it fires, the more potassium builds up. So if it fires too fast, it might not be able to maintain that firing. So we asked, what's the maximum frequency a cell can fire given conditions for the blood flow, how much oxygen and ATP there is? And actually, the question that initiated this project, the surgeon asked me was, do heterogeneous capillary flow patterns result in lower brain tissue partial oxygen pressures? By the way, I use the phrase partial oxygen pressure throughout the talk. Partial pressure, that's just very closely related to the oxygen concentration. So if I say partial pressure, it just means really the concentration of oxygen. So there's a number of studies lately that have indicated that as we get older, perhaps, our properties of blood flow, like diameters or response to signals or whatever, becomes more heterogeneous. And these heterogeneities can result in lower oxygen levels, and that could lead to various diseases. So an outline of my talk is I'm going to consider the three components separately models. So I'm going to throw a lot of equations at you, but I'll just try to highlight what's important for this talk. So I'll talk about each component separately, and then I'll try to couple them all together. So I think here's the neuron. So here's a lot of equations. This, I think you're the most familiar with you have the usual membrane equation. Of course, what's different between this and the Hodgkin-Huxley is that the Nernst potentials are not constant. They depend on concentrations of sodium and potassium. So we need equations for sodium and potassium inside and outside the cell. You think I can do this here? Yeah, that term there corresponds to the uptake of potassium by astrocytes. So, you know, astrocytes is one of the main things that buffers extracellular potassium. So in that equation, the astrocyte membrane potential VA is just assumed to be constant. And there's an equation for the pump. Let's see if I can do that. Where is the, there it is, the pump. The pump depends on, I threw in a term that you usually don't see related to ATP there. So for now, I'm going to assume that ATP is constant. And then when I talk about the mitochondria model, then ATP will, you know, change. But for now, let's assume it's constant. And I'm going to have an excitatory periodic input to the cell at different frequencies. I'm going to ask, what is the largest frequency that the cell can maintain firing? So here, I just, like, for example, in this panel, the ATP level is at some level, I lost this thing, whoops. And the frequency, well, 4 hertz, and GKA is 0. That means there's no astrocyte. And the cell has no problem maintaining firing at that frequency. If you go to the middle panel, I lower the ATP levels from 1 to 0.1. And the cell goes into depolarization block. There it's still at 4 hertz. In the right panel, it's at 10 hertz. But I added the astrocyte, and it has no problem maintaining the firing at 10 hertz. Okay, so that's the neuron model. Now for the mitochondria. This is the based on the Magnus-Kaiser model for ATP production. And again, there's lots of equations and stuff. The important thing to know is that it depends on oxygen, and it produces ATP, okay? And so now, PA, in that red box there, that's PAVJO, that's the, PAV is the average oxygen partial pressure in the surrounding tissue. So some tissue surrounding the neuron, that's the average amount of oxygen. And I'm assuming that's constant now, okay? That has to change when I introduce the blood flow model, but for now it's constant. Now, for us, something that's going to be very important is the oxygen consumption rate. At what rate is oxygen being consumed? And that's the term gamma PAV JO. JO is basically the consumption rate. But if the tissue level is too low, then that goes down. So you multiply it by that's gamma PAV. So that's the we'll need the oxygen consumption rate later in the talk. And notice that this depends on calcium. There's an equation for calcium in there. And you would think that there's some way for the neuron to tell the mitochondria what its energy needs are. So if it's firing faster, maybe it should produce more ATP. So the way I put that in the model is I assume that kind of the intracellular cytosolic calcium goes up if it fires more. So what I do is I add the so-called Lee-Rinzel model for cytosolic calcium. And in that model, there's one of the variables is IP3. So in my model, IP3 is a parameter. And if you increase IP3, that will increase the amount of calcium coming out of the endoplasmic reticulum, and that will increase the ATP production. So the way the neuron communicates its energy needs to the mitochondria is through this parameter IP3, but I do not explicitly model how that's done. I just assume it's a parameter. Okay, and so, I use XPP to solve these equations. On the left there, you see the response to different levels of IP3. If IP3 is high enough, 0.2, then you get these nice calcium oscillations. And if IP3 is low, then calcium stays low. And you can also plot the consumption rates. Like I say, the consumption rate is going to be very important. And you can see how the consumption rate and ATP production depends on, for PAV, which is the average tissue oxygen levels, or IP3. So if the tissue levels are low or IP3 is low, then the consumption rate is low. And if they're high enough, then the consumption rate goes to some elevated level. All right, so now I want to start combining the model. So how do we combine the neuron and the more Magnus-Kaiser model? Well, they're coupled through the cytosolic ATP levels. So in the previous slide, I didn't have anything related to the neuron, but so what we do is we add a term corresponding to the pump. So the pump is going to consume ATP. So the equation for ATPI depends on the mitochondrial production, which is this term in here, and the consumption by the pump. And one can actually analyze that mathematically using fast-slow analysis. So I'll just flash a few slides here of panels. The slow variables are the extracellular potassium and the cytosolic ATP levels. So if you fix ATP and you draw the bifurcation diagram, you think of Ke as a slow variable. So if it's constant, then you get a nice bifurcation diagram like that with a homoclinic orbit and things like that. And down here, this curve is, so here at ATP, so for different values of ATPI, the homoclinic orbit changes. So this is the curve, if I can get it, of homoclinic orbits. And if you project the variables Ke and ATPI onto this thing, you see that you get depolarization block whenever you cross this homoclinic orbit. So using that, And well, you know, it's a bit of a work. Yeah, I construct a reduced model and et cetera, et cetera. But one can compute how the maximum frequency that the neuron can respond depends on the average tissue oxygen level, PAV. So here I'm showing how the maximum frequency that the neuron can respond before it goes into depolarization block depends on either PAV, which was the tissue oxygen levels, or this parameter IP3. And I consider different cases with astrocytes or without astrocytes. With astrocytes, it can, this model can fire, like 50 hertz. And but without astrocytes, it can't go really above like 20 hertz. Okay. So now the last piece of the puzzle is blood flow. So more equations. So there's a capillary and there's the tissue. So I use, omega B is the partial pressure in the capillary, and it satisfies some ordinary differential equation, because I think of the capillary as a one-dimensional table. And in the tissue, this is steady state, okay? Steady state condition. You have a Laplacian of omega, so you have diffusion. And this is an important term. This is in here. This is the consumption of oxygen in the tissue. So if you increase the consumption in the tissue, you're going to have less oxygen. So the important parameters here are Q. That's the flow rate. That's how fast, how much oxygen is flowing through the capillary. So if Q goes up, you get more oxygen and presumably more ATP. MT is the consumption rate. Rate is the oxygen being consumed in the tissue. And then you need a boundary condition. So that's the boundary condition, okay? So in the remainder of the talk, I'm going to consider a simple capillary network. There's just 4 vessels. Blood is flowing to the right. And there's this nice software by Seacom that we use to solve the equations for blood flow. And what you can do is with this software is for different consumption rates, you can figure out what the tissue levels are. And what I do is I can consider the tissue levels averaged over where the blood is leaving this region. So each one of these curves tells me if you increase the consumption rate, then you're going to have less oxygen. So PAV goes down, and you can do that for different flow rates. So obviously, well, I don't know if it's obvious, but if you increase the flow rate, if more oxygen is entering the system network, then you're going to have more oxygen. So that's what that curve means. So now I want to combine the models. So how am I going to combine the models? Well, what I'm going to do is I'm going to assume that the rate at which oxygen is being consumed in the tissue is exactly the same as it's being consumed in the mitochondria. I mean, that's why oxygen is being consumed, because it's being used by the mitochondria to produce ATP. And I don't know if you remember what all these terms mean, but it means that these are the various consumption rates. But anyway, this gives rise to a map. The map says that if you tell me what the consumption rate is in the blood, from the previous slide, you can compute the average oxygen partial pressure. And from a few slides ago, I showed you that if you know what the average partial pressure is, In the tissue, you can compute the, well, I have JO, but that's related to the consumption rate of the mitochondria. And so what we want to do is we want to find a fixed point of this map. So we just use MATLAB to do that. So this is kind of a busy picture, but what it shows is, see, if you tell me what PAV is, the average tissue oxygen, these various curves here are the consumption rate in the mitochondria. And there's different cases. There's with astrocytes are in black, without astrocytes are in red, and dashed is, and then I have different levels of IP3. On the other hand, if you tell me what the consumption rate in the blood is, in a previous slide, I said you can compute what the average tissue level is. So, and I do this for different flow rates. And the thing is, where these curves intersect is where the fixed points are. So what this says is if you tell me what the flow rate is, this is the combined model, I can tell you what the average tissue oxygen levels are, partial pressures are. This combines the consumption of oxygen both in the mitochondria and the tissue, and we have the different cases of with astrocytes and without astrocytes and different levels of IP3. So, and then a number of slides ago, I showed you that if you know what the value of PAV is, the tissue level, you can compute the maximum frequency. That's where I use that bifurcation diagram. So what I now have is if you tell me what the flow rate, that Q in the blood is, I can tell you the maximum frequency that the neuron can fire before it goes into depolarization block. And my last slide is the issue of homogeneous versus heterogeneous networks. So if let's assume that the total blood flow rate is Q for all 4 vessels. The homogeneous case is if all the flow rates of each of the vessels are the same, so Q over 4. On the other hand, the heterogeneous case is basically you choose random numbers, so the flow rates add up to Q. All right. So that, heterogeneous flow rates. And what this curve shows is the dashed is heterogeneous, the solid is homogeneous. Sorry, I'm not doing too good. For different values of the flow rate. But anyway, this, if you look at the black curves, the dashed is below the solid. So that means the heterogeneous case produces less oxygen for a given consumption rate than the homogeneous case. So it shows heterogeneities lower the average oxygen levels. And, oh no, this is my last slide. So then we ask the following question. For which values of Q, that's the blood flow rate, and IP3, that's ATP production, is the average oxygen level above some threshold? So the idea is that this threshold, surgeons or doctors sometimes say that what is, the pathological, when is the patient, may have a stroke. And you say if the average level, oxygen levels get below some level, which I call P naught. So the question is how fast must the flow rate be? And IP3 is, the ability of the neuron to communicate its energy needs to get above, so you guarantee that the oxygen levels are high enough. So using this analysis, we're able to draw a picture like this. So there's IP3 levels, there's the minimum flow rate, and there's different cases with astrocytes and without. So black is with astrocytes. And notice the dash curve, black curve, is above the solid black curve. So that means that if there's heterogeneity, you have to have a larger flow rate. You need more oxygen entering the system in order to keep the tissue oxygen levels above some threshold. And actually, that's my last slide. I didn't get around to doing a discussion. I guess as far as discussion, for me, it was just a question of, is one able to combine this model and compute how, the neurons firing depends on the flow rate and energy needs in a somewhat realistic model. I like to emphasize that even though I had zillions of equations and things, it looks complicated, that this is in some sense a minimal model. There's many, many, many things that are unknown and not included in the model. For example, how does the neuron communicate with the blood? Presumably as it needs more oxygen, that's going to communicate to the blood to dilate or increase the diameter or whatever. Those are really just parameters in our model, but they're very controversial how all that happens. So I did not explicitly model it. And I guess with that, I'll stop.
Thanks, Dave.
Should I stop the share?
No, let's hold on for a minute. So at the moment, the chat is empty, but so people could either type questions in the chat, or I'm trying to look over the participant list. So if you prefer, you can sort of raise your hand in the participant list if you have a question. And if you can't manage either of those and it happens to be quiet, then you could just unmute and jump in. So do we have questions? I can start with one. In reality, I guess there would be interaction of the blood flow with the astrocytes as well. Is that in the model at all, or is that something to think about?
I didn't really say that. There's a bunch of details I didn't say. In what I have, I just have that the consumption rate by the astrocytes is pretty much constant. It's that gamma function. But you're absolutely right there. The astrocytes consume energy, and that could depend on flow rate too, and things like that. So that maybe can be modeled in a similar way. But I just, in this simple model, I just have it basically be at a constant level.
So I guess with heterogeneity, with that additional detail on the astrocytes, it could get even worse. Like,
oh yeah.
Have even, you need even more flow. Okay.
There's communication, of course, between the astrocytes and the blood, and
okay, so they can
that's where the communication probably takes place anyway. So
okay, nice. All right. Are there any other questions? Okay, yeah, here we go. So a question from Peter Thomas. When you were setting up the fast-slow analysis, how did you decide which variables are slow in this particular model?
It turns out that you don't really even need most of the mitochondrial model for it. So I think it's natural to have extracellular potassium, and then for the neuron, there's conservations of potassium and sodium. So, and if you use some electrode neutrality, you can basically get rid of the other ionic variables. So you're just really left with potassium, ATP, and then the memory potential and the recovery variable. So then it's kind of natural just to have those two variables, if that answers it. Okay, the first step is to reduce the model to a simpler one, and then it kind of pops out that those are the natural variables to choose.
Okay. And one final question before we go to the next speaker. This is from Mohammed Abdullah. can these kinds of models account for delays between neurons, monitoring, signaling for, and satisfying their energy needs? And I guess more importantly, would this have significant impact on the network behavior? The delays.
I guess I wouldn't, explicitly throw delays in there, but I guess you can add processes that naturally, that have different time scales and take certain times to get above certain thresholds, and that would naturally introduce delays. And so that's, or you get propagation along neurons or whatever. So that's how I would introduce delays, I suppose.
Okay. So as far as the impact, if you want to follow up on that in the chat, feel free to type, but we should move on to the next talk. So thanks, Dave.
Thank you.
All right. So you can stop sharing and maybe go ahead and mute. And our next speaker is Ryan Phillips. So Ryan, can you share?
Yep. 1 second.
While Ryan's sharing, I'll mention Ryan is just finishing up a postdoc with me and moving on to a position in the University of Washington in Seattle. So I think he's sitting there sweating in Seattle at the moment, and talking about the effect of chloride dynamics on substantia nigra parasiticulatum responses to palatal and striatal inputs. Go ahead, Ryan.
Thank you. Yeah, it's really hot here in Seattle. It's supposed to be 105 today. All right, so I'm going to talk about the effects of chloride dynamics, as John said, on substantia nigra parasiticulatum responses to palatal and striatal inputs. All right, so first off, what is the SNR? So the SNR is a nucleus within the basal ganglia. We're focusing on just in the SNR first. The SNR is a collection of neurons that are tonically active, and they provide tonic inhibitory input to downstream motor centers. So if you can get some sort of pause or substantial depression in the firing rate of these SNR neurons, then you can gate motor actions such as reaching, walking, or saccade. Within the basal ganglia, there are two different pathways that regulate SNR activity. So there's the indirect pathway and the direct pathway. In the direct pathway, if you were to stimulate these D1 neurons within the striatum, they provide inhibition to the SNR. So shown here, if you stimulate, you're going to inhibit the SNR, you're going to decrease the inhibitory output to motor centers, and you'll facilitate motor actions. In contrast with the indirect pathway, if you stimulate the D2 neurons, then you inhibit these GPE neurons, which disinhibits the SNR, but it also disinhibits the STN, which provides an increase in excitation to the SNR. So the net effect on the SNR is disinhibition plus excitation, which will increase the inhibition of motor centers and suppress motor output. And this is what we see in experiments. So experimentally, if you optogenically stimulate the D1 neurons of the striatum, you get this increase in firing rate. And the average response within the SNR is this decrease in firing rate. And you get a corresponding increase or facilitation of movement. And then the indirect pathway, as you predict from the model up here, you stimulate the indirect pathway of the D2 neurons right here in the striatum. You get increase in firing rate in those neurons. And you actually get some excitation or disinhibition of SNR neurons. And as you predict, you see a suppression of motor movement. And so this all seems to check out at this high level population average viewpoint. But if you look a little bit closer at what's happening within the SNR neurons, you actually get a lot of neurons that are inhibited and excited, in this case with direct pathway stimulation. And with the indirect pathway, it's kind of the same story. The average effect here in the SNR is excitation, but you have some neurons that are excited and you have a lot of neurons that are inhibited. And you also have a substantial number of neurons that have no effect. All right, so then the question is, can these diverse responses be driven by chloride regulation or chloride dynamics that can result in hyperpolarized or depolarized E-GABA? All Right, so what I didn't quite highlight yet is that these inputs into the SNR from the D1 or the direct pathway and indirect pathway, these are GABAergic synapses. And typically you think of GABAergic synapses as being inhibitory, which usually they are, but it's also known that they can be depolarizing. And this depends on the chloride, the intracellular chloride concentration. And to get a better understanding about that, talk a little bit about the basics of GABAergic synaptic transmission. So GABA A receptors are permeable to both chloride ions and HCO3. And together, the reversal potential for these two separate ions make up the GABAergic reversal potential by the Goldman equation here. So typically, the HCO3 reversal potential is -20. The chloride reversal potential is -90. And then there's a 4-to-1 ratio of chloride to HCO3 that fluxes through the channel. And so together, that sets up a reversal potential of usually close to -65, which is just below the after hyperpolarization after neuron spikes. All right, so what are the factors that regulate the intracellular chloride? So it's very simple. It's the balance of how much chloride comes in and the chloride load versus how fast can the cell extrude chloride through the KCC2 pump. And then another thing that can happen is, so based on the balance of this chloride extrusion capacity and chloride influx, you're going to set the intracellular chloride concentration, which in turn will set E-GABA and determine on whether or not the GABAergic reversal potential is inhibitory or excitatory, or potentially shunting. And then also, if you get a very large transient chloride load, you can actually get a biphasic response, where initially you get strong inhibition, but due to the substantial chloride influx, you actually get a hyperpolarization of the chloride reversal potential, which can actually shift the GABAergic reversal potential to become depolarizing. And that's how you can get an inhibitory to excitatory effect. All right, so when are chloride dynamics important? So the first one is if you have a large chloride load, a tonically high chloride load. So this can shift and overcome the chloride pump and shift the intracellular chloride concentration to higher levels, which can give you potentially depolarized E-GABA. And also a small compartment size. So you have a very small compartment size and you get a transiently high chloride input. On relatively short timescales, you can get short-term ionic plasticity where you get this large chloride influx and then a fairly fast increase in the intracellular chloride load, which can give the biphasic responses that we saw in the last slide. All right, so let's look back in the SNR. And what do we have in the SNR? So we have converging GABAergic inputs to the SNR. If you look a bit closer at the actual cell bodies, these two different pathways form synapses on different locations of that SNR neuron. So the indirect pathway GPE neurons, they form synapses on the soma, whereas direct pathway neurons form synapses out on the distal dendrites. So GPE neurons outnumber SNR neurons and they broadly project to the SNR, and GPE neurons fire at a high tonic firing rate. So this is going to provide a high chloride load to the soma of SNR neurons. And out in the distal dendrite, as you expect, the compartment size is very small. So within the SNR, you have the potential for both depolarized E-GABA from large tonic chloride load, and you also have the potential for short-term ionic plasticity due to the compartment size. All right, so the outline for this, I already covered the introduction. So the first question I'm going to try to ask is, can chloride dynamics explain and predict the diversity of SNR responses to indirect and direct pathway stimulation? So I'm first going to introduce the SNR model, simulate SNR responses, and then compare some experimental data. And so obviously the answer to this first question is yes. So after that, after we go through this part two, we're going to go into the last part of the talk and talk about some functional implications, such as local SNR synchrony and oscillations, and if we have time, perceptual decision-making. All right, so we have a two-compartment model for the SNR. We have the soma with the GPE inputs, and we have the dendritic compartment with the striatal inputs. And these are the simple voltage equations that govern the voltage dynamics of the two compartments, which are coupled together. And then we tune the model to meet some basic experimental benchmarks, such as baseline firing rate, the magnitude of the after hyperpolarization, and FI curve. And of course, we have chloride dynamics governed by these two this differential equation, and then the Goldman equation for calculating the GABAergic reversal potential. All right, so predictions of the model to GPE and striatal stimulation. So to get into this, first with the thing about the chloride dynamics, and again, as we talked about before, it has to do, the GABAergic reversal potential will be set by the balance of the chloride influx and the rate of chloride extrusion. So on the x-axis here in the 2D plot, we have the tonic chloride load into the soma, and we have the extrusion capacity. So depending on where you sit in this 2D space, you're gonna have different GABAergic reversal potentials. So on average for a low extrusion capacity, you're gonna tend towards more depolarized E-GABA. And as you decrease the chloride load, you're gonna tend towards more hyperpolarized and more inhibitory GABAergic reversal potentials. All right, so with GPE stimulation, so this is the one that goes onto the soma, if we pick different spots within this 2D space, we can get all kinds of different responses. So up here, this point B, we have a pretty low chloride load, but strong extrusion capacity. If you stimulate these GPE synapses, we predict you can get complete inhibition. If you depolarize E-GABA a bit, this point C here, where you're in a kind of a shunting regime, you can have no effect on the firing rate. I think you affect the height of the action potential a little bit. And then as you expect, as you go to a depolarized E-GABA, you get excitation. And then we have two different types of what we're calling partial inhibition. The first one is just kind of in a regime in between the no effect and complete inhibition, where in this case, you just have a slowing of the firing rate. And then the second type of partial inhibition, you actually start off in the complete inhibition state, but due to chloride accumulation, you depolarize E-GABA and you can recover firing a little bit. All right, so the same story with the striatal inputs. So you have the same range of E-GABA, and we also get complete inhibition, no effect excitation, and two types of partial inhibition. This type of partial inhibition over here, the E2, is again an effect of a gradual chloride accumulation that lets you escape from inhibition. The other type of partial inhibition you see here, you start off just slowing the firing rate, but then you transition into complete inhibition. And this is an effect of some of the short-term synaptic dynamics that I didn't have a chance to talk about, but the striatal inputs they undergo short-term facilitation. So the synapses are actually becoming stronger during the stimulation period in this case. All right, so then the new response we see with the striatal stimulation is that you can actually get a more extreme case of this second partial inhibition, where you get really rapid accumulation of chloride. And in this case, we would predict that you can get a biphasic inhibitory to excitatory response. So you start off with really strong inhibition, which transitions into excitation. All right, so let's compare this with experimental data. So with the collaborators at CMU, Aaron Geddes, we collected some recordings to see, to test our model predictions. And so what we see is that we get all the different types of response types that we would predict. And surprisingly, we actually even see some biphasic response with the GPE stimulation. The ratios between of the responses are very different for these two, as you'd expect. So with GPE stimulation, we do see a couple biphasic responses, but there are very few of them. Mostly what we see is partial inhibition and some excitation, but very few neurons are actually completely inhibited. Whereas with striatal stimulation, we see a lot of neurons that are initially strongly inhibited, but we also see way more of these biphasic responses, which indicates, as you expect, that in the striatal or in the distal dendrite, where you have a small compartment size, you have more likelihood to have a fast chloride accumulation. And then we also have quite a few more excitatory responses in the GPE case, which you'd expect with the large tonic input from the GPE onto the SNR. All right, so the key points to take away from here is that E-GABA and the SNR neurons are not purely inhibitory, and that E-GABA can dynamically change in the order of seconds. All right, so the first answer to the first question in the talk was is yes, that chloride dynamics are important within the SNR. And so next we want to look at some of the potential implications of this. the first one is local SNR synchrony and oscillations. All right, so within the SNR, there are local collaterals, although they're very sparse. On average, an SNR neuron receives 1 to 4 inputs from a neighboring SNR neuron, but they do exist. So there's an example shown here of a connected pair in a feed-forward connection. So what we want to ask now is, can changing E-GABA affect the synchrony of connected pairs within the SNR? And to do this, we're going to use phase response curves. So a phase response curve characterizes how a stimulus at a certain point in the phase can advance or delay the onset of the next spike. So in this example here, if a stimulus happens in the middle of the phase, so 0.15, and we have a hyperpolarized E-GABA. So what this does is causes a delay. Whereas if we have a depolarized E-GABA and we stimulate the same point, we'd actually get an advance. All right, so what we did is calculate or quantify the phase response curves. So these curves are characterized the advance or delay that happen as a function of the stimulus phase here. And we did this for E-GABA from -50 to -60. So you take the first case of -60, what you see is that no matter where you stimulate within the phase, you always get a delay. But this delay peaks at roughly about 0.75. And then as you depolarize E-GABA, then you start to dip into some advances. And so the case of -50, you're almost always going to advance the next phase until you get to about 0.8, where it transitions back to a delay. All right, so we can use these phase response curves to predict the synchrony within the SNR. So we're taking this first curve here at -60. It's blue now. But if you look at where this curve crosses the x-axis, you get a fixed point. In this first case, so this is we're going to use this phase response curve to predict the phase relationship between two neurons in a unidirectional feed-forward connectivity pattern. In this case, you would expect a fixed point roughly at synchrony here, but this is unstable. So the histogram right here, which shows the timing of the input to this postsynaptic neuron, shows that you can have a pretty wide spread of the synchrony here, or the relationship. However, it does tend to stay close to synchrony. So as we move to more depolarized cases, so in this case -55, then you actually get a stable fixed point, and you get stable phase locking here, indicated by this histogram. And you move farther to -55, and the phase locking moves over to 0.8, but again, it is stable, indicated by the histogram here. All right, so if we put this all together and create a histogram from -60 to -55 for E-GABA, we can actually predict how the synchrony in this feed-forward pair would change as a function of E-GABA. So for very depolarized E-GABA, you get pretty close to synchrony, but then as you hyperpolarize E-GABA, you shift towards being antiphase or less synchronous. And then as you become more and more depolarized, you tend towards synchrony again. All of this fixed point becomes unstable, and so the histogram of the spike times spreads out. All right, so if we take a little bit of a closer look at what's happening over here on the left-hand side for hyperpolarized E-GABA, when we have these unstable fixed points, is we actually get oscillations. And these oscillations show up as transient dips in the firing rate of the postsynaptic neuron. These happen at roughly 1 to 2 hertz. All right, so another way to look at these oscillations is in the input phase. And so this is the phase of when the presynaptic neuron fires relative to the phase of the postsynaptic neuron. And you can see this really nice, clear oscillation in the input phase as a function of time. And so using this, you can understand, so using this and the phase response curve, you can understand how this oscillation occurs. These are phase slipping oscillations. So in the case where we have this unstable fixed point, if you pick a point here, so we start off at 1, so you're roughly synchronous, you're close to the fixed point, but the phase response curve predicts that you get a delay when you're here. So you go up and you get a delay, and you get kicked back in the cycle a little bit, and then you get a bigger delay, and you can go through the cycle and you get bigger and bigger delays, and you shift your phase relationship until you get down here to where the phase response curve is very small, and then you start getting very small changes in the phase progression, and you get stuck relatively close to synchrony until you get kicked around the other side and the cycle repeats again. All right. So one thing that, so the shape of this phase response curve is highly dependent on the reversal potential. So if you were to increase the, or hyperpolarize the reversal potential more, the peak of the phase response curve is going to grow, and you're going to get larger and larger delays. So the effect of this is that you're going to get bigger kicks that are going to delay your progression even more, and you'll go through the cycle faster and faster. And so if you change E GABA, this is exactly what you see. So at roughly minus 58, you start out with oscillations at roughly at 1 hertz. And if you hyperpolarize E GABA, you can get up to roughly 3 hertz for these types of oscillations. All right, so these oscillations that are shown here occur in 2 identical neurons, which isn't necessarily a very realistic case. So we did check to make sure that these types of things can happen in a wide array of settings. So we changed the firing rates between the 2 neurons, increased the overall firing rate, added Gaussian noise, synaptic delay, looked at multiple presynaptic neurons and some 3-neuron motifs. And the general conclusion is that these types of oscillations are very robust to all these different perturbations. I don't have time to go into all the details of these, but happy to answer questions about them after the talk if there's time. All right, so another test of how robust this is was to see if we still get these oscillations in a larger network of the SNR. So I constructed a 100-neuron SNR network that's heterogeneous with random connectivity. So the connectivity profile is shown here with this histogram with the number of, so there's number of connections and the number of neurons with those connections. So the average number of connections or inputs to a given SNR neuron is 2. And as you change E GABA within this network, you change the firing rate. So low E GABA, you get a lower firing rate, increased CV, and higher E GABA, you get an increase in firing rate, and in general, a decrease in the CV. That is the effect of the local SNR collaterals. All right, so to cut to the chase, with depolarized E GABA, we do still see these slow oscillations. I've highlighted a couple shown here. So this 2D plot here shows the cell ID and then the power spectrum. And you see some peaks here at close to 1 hertz. And so I pulled out these 2 examples here. So cell 46 and 18, and we get these nice peaks in the power spectrum. And if you look at the frequencies, sorry about that. So yeah, if you look at the frequency for these different neurons, you get these nice oscillations in the frequency. One surprise is that for very depolarized or excited for E GABA, we actually start to see in the larger network slow oscillations that occur too, which was a bit of a surprise. All right, so the key points from this part on SNR synchrony and oscillations is, so far what we have is that the chloride load to the SNR can change E GABA, which in turn can change or regulate the SNR synchrony and the emergence of these slow oscillations. All right, so are there implications or and some experimental comparisons we can make? So yes. So in dopamine depletion, the classic response is that the firing rate of the GPE neurons actually decreases. And from our model, you would predict that if you decrease the firing of the GPE, you're going to decrease the chloride load to the SNR neurons, and you should hyperpolarize the E GABA. So you get a shift in the chloride load, which will shift E GABA and shift you from potentially asynchronous state to synchronous state with some oscillations. And this is exactly what we see in the experiments. So in dopamine depletion, you see an increase in the synchronous pairs within the SNR, and you see the emergence of these slow oscillations. The next question is, can you counteract these? So can you optogenically stimulate the GPE to increase the tonic load back to the SNR, which you would predict from our model, you should shift this back up in the chloride load and increase E GABA, and that should shift you on this curve, so you should decrease synchrony and remove the oscillations. So we did that experiment. So we identified some of these slow oscillatory units within the SNR, shown here, identified by the power spectrum. And then we optogenically stimulated GPE neurons. And one surprising thing is that you actually get no change in the firing rate of SNR neurons, but you get a substantial reduction in the oscillations of these neurons. All right, so key points to summarize this. So the main idea is that the GPE to SNR projections provide a tonic chloride load that tunes SNR E-GABA, and through local collaterals within the SNR, this E-GABA can regulate the local synchrony and the emergence of these slow oscillations. All right, so finally the last thing is decision making and dendritic chloride regulation. All right, so a paradigm for decision making is a perceptual decision making task where an animal looks at a screen with dots that are moving different directions, and their task is to decide which way on average these dots are moving. And within the SNR, what you see from experiments is that this is a model here, but you see a ramp in activity in the direct pathway for neurons that are trying to gate a left versus right decision. And once you accumulate enough evidence in this ramp in the striatal input, then eventually what you get is a nonlinear and abrupt pause in the SNR activity that can gate a saccade to look left or right. And so with our idea here that the GP input to the SNR can tune E-GABA, we would expect the same thing with a dendrite. So this first panel right here, and see if you, or I guess back up a little bit, we brought back the SNR network, which has a mean firing rate of roughly 25 hertz. And then the first thing we did is we changed the tonic input to the soma and looked at whether or not we can influence the dendritic E-GABA. And yes, you can. So as you increase the chloride load in the soma, you depolarize the somatic and the dendritic reversal potential for GABA. So next, what we did is we constructed a ramp in the firing rate of the striatal synapses for these neurons, and then we characterized how the chloride load affects how long it takes for the SNR activity to decrease below some sort of threshold to generate a pause. And what we found is that as you increase the tonic chloride load, as you expect, you're going to weaken striatal synapses on the SNR, and it's going to take longer for you to pause in this type of paradigm. All right, so then to conclude, again, so the GPE to SNR synapses provide tonic chloride load to the SNR neurons. The tonic chloride load tunes somatic and dendritic E-GABA. And then E-GABA can regulate the local SNR synchrony, the emergence of the slow oscillations. And then finally, E-GABA can also tune the response times within the SNR with it for decision-making tasks. All right, and I'd like to thank John Rubin, my advisor at Pitt, and then also collaborators at Carnegie Mellon, Aaron Giddis, Ian, and then Tim. All right. I'll stop there.
Thanks, Ryan.
Yep.
So we have a short amount of time for questions. Again, questions can go in the chat. Hands can be raised, or as a backup, you can just unmute. So does anyone have a question for Ryan? Just gonna give a minute. People can gather their thoughts. And if not, we'll move on to the next talk. Going once.
Yeah, I'll ask a question.
Okay. Bill Meyer, go ahead.
Yeah, so your talk is mainly focused on chlorides, and I like it. So great talk, thanks for that. But I was wondering about the bicarbonate. That seems to play less of a role in your talk, but the reversal potential, the formula you give for it, depends on it. So what's the motivation? Because if this bicarbonate quantity is large, then chloride hardly has an influence.
Right. So yes, if you could shift the bicarb, then yes, you could affect the GABAergic reversal potential as well. In the intracellular space, the bicarbonate concentration is regulated by a really fast, I think, catalytic reaction. And so in general, it's really hard to change the intracellular bicarb. I think there might be some diseases or some cases where you can affect the extracellular bicarbonate. but that's not really in the context of dopamine depletion or healthy animals, I don't believe. So in general, you can assume the bicarb to be fixed.
Okay, thanks. So actually, we're down to sort of the last minute here. So Ryan, why don't you go ahead and stop sharing? Great. And Louisiane, you can share.
Yes.
Thanks again, Ryan. So I'm pleased to welcome our next speaker, Louisiane Lemaire from INRIA, somewhere down in southern France, I think. And she'll be speaking about another form of ionic channel that is impacting neuronal dynamics in migraine and epilepsy scenarios. So Louisiane, please go ahead.
Okay, so thanks for giving me the opportunity to present my work today. We are working on the mutations of the sodium channel called NAV 1.1. And it happens that some mutations of this channel cause migraine, while other mutations cause epilepsy. So we are studying those mutations with a modeling approach. And we are working in collaboration with neurophysiologists from the IPMC lab who are doing experiments on mice, in particular with Massimo Mantegazza. I will first present this sodium channel and its mutations, and then the model which we are using to study them, and finally the results concerning the migraine mutation and the results for the epilepsy mutation. So NAV 1.1, it's a voltage-gated sodium channel, which is mainly expressed in GABAergic neurons, where it is very important for their excitability. And several mutations of this channel have been identified, which cause familial hemiplegic migraine type 3, FHM3. And this is a subtype of migraine with aura. So there are sensory disturbances before or during the headache. And the common point of all those migraine mutations of NAV 1.1 is that they cause a gain of function of the channel. So the activity of the channel is enhanced. What is interesting is that other mutations of the same channel cause epilepsy, for instance, Dravet syndrome, which is a severe childhood epilepsy. And those epilepsy mutations of NAV 1.1 are loss of function mutation. And in both cases, the link between the mutation and the disease is not completely clear. In the case of the migraine mutations, it is thought that what causes the headache is cortical spreading depression, CSD. It's a wave which slowly propagates in the cortex. And here you can see on the left the recording of the voltage of a pyramidal neuron during CSD. There is an intense firing frequency and then a depolarization block. It's this plateau here at a voltage which is much larger than the resting potential. And this wave slowly propagates in the cortex. You can see on the right the spatial propagation in white. Those are a picture of a brain slice of a mouse. And the two recordings are from our collaborators. And it is thought that when this wave reaches the meninges, it will indirectly stimulate some pain receptors, which will cause the headache. So there are some ideas concerning the link between CSD and the symptoms of migraine, but what is not so clear is the link between the mutation and CSD. And what interests us here is rather the initiation of CSD than its spatial propagation. This is our model. So we consider a pair of neurons. There is a pyramidal neuron, which is excitatory, and a GABAergic neuron, which is inhibitory. And we take into account all the different transporters, which you can see in this diagram. So like for Hodgkin-Huxley model, we have, for example, sodium and potassium voltage-gated channel. We also have leak currents, but we have other transporters, which are important for what we want to study, for example, sodium-potassium pump for restoring ionic gradients. And this model is based on a model developed by people in my team, but we made several changes to adapt it to the situation. So first of all, we model the mutations of NAV 1.1. And since this channel is mainly expressed in GABAergic neurons, in our model, the mutations, they will only affect the GABAergic neuron. And I will explain later how we model the mutations. Something very important is that we take into account the dynamics of ion concentrations, which are often assumed to be constant, and this is reasonable in a normal situation. But during epilepsy or migraine attacks, the ionic gradient, they are completely disrupted, so we don't make this assumption. And as a consequence, the two neurons, they are not only coupled through synapses, but also through the variations of the extracellular ion concentrations, we have one extracellular ion concentration per ion. And finally, we changed the model which is used to describe the opening and closing dynamics of the voltage-gated channels of the GABAergic neuron. Why we did this? It's because our collaborators, they are making some experiments where they use a toxin to mimic the effects of the migraine mutations of NAV 1.1. And when they use this toxin, they observe spontaneous CSD only in the neocortex and not in the other brain regions. So it was important for us to have a cortical model for the GABAergic neuron. In our model, this is how we model the mutations. So NAV 1.1, it's a voltage-gated sodium channel, which is so it's closed at the resting potential. And if the voltage increases, it opens. But there is another mechanism which makes it close even though the voltage is still high. This is the inactivation mechanism. And those activation and inactivation mechanisms, they generate a transient sodium current, which we model with this expression here. This variable H represents the inactivation. And a common point of most migraine mutations of NAV 1.1 is that it will cause persistent sodium current. The inactivation mechanism is affected. And this is how we model persistent current. You can see that the variable which models the inactivation here is always equal to 1, so there is no inactivation. To model the migraine mutations, we partially replace the transient current with persistent current. And so the parameter which we increase is this percentage here, representing the relative importance of the persistent current. To model the epilepsy mutations, so those are loss of function mutations. So what we do is simply we reduce the maximal conductance of the transient sodium current. In our model, there are some conserved quantities, and this is a direct consequence of the fact that we have dynamic ion concentrations. Each ion current appears both in the equation for the membrane potential and in the equation for the corresponding intracellular ion concentration. And because of this, the quantity here, it's a first integral for our system. It's constant over time. So because of this, we do not model external inputs to the neurons using a constant parameter on the right-hand side of the equation for the membrane potential. Because if we would do this, then instead of having something constant here, it would increase with time, and so we could not have limit cycles or steady states. So we don't do like this. Instead, we model external inputs to the neurons with an excitatory synaptic input on the AMPA receptors, which are this one and this one. And we can decompose it into sodium and potassium components, which will appear in the equations for the sodium and potassium ion concentrations. So we don't have a drift of the system. And when we want more or less stimulation of the neurons, we modify this conductance here. Okay, so this is the equations of the system. I will not enter into the details, it's just to give an overview. And using the first integrals, which I mentioned, we remove two equations. Okay, now I will show the results. So first, the results concerning the migraine mutations. first, we focus on the effects of the mutation on the firing properties of the GABAergic neuron itself when it's not connected to the pyramidal neuron. We compute some input-output relationship. So we start from the neuron at rest, and we give it some external input, and we count the number of action potentials which are generated in a given duration. And we do this for different values of persistent currents. High values of persistent currents model the mutation, and otherwise is the control condition. And we don't really see an effect of the persistent currents on the firing frequency of the GABAergic neuron. What we see, it's an effect on the extracellular ion concentrations. When we have more persistent current, the firing of the neuron releases more potassium in the extracellular space and uptakes more sodium. This is possible even though the firing frequency is similar, because the ion fluxes at each action potential are increased when we have, so this is a single action potential. And when we have persistent sodium currents, we have more potassium currents and more sodium currents. What is the consequence on CSD initiation in our model? So migraine, it's an episodic disorder. So there are symptoms only during the attacks, and otherwise physiologically everything looks normal. And this means that there is a trigger factor to cause the transition from the healthy state to the pathological state. And in our model, persistent sodium current reduces the threshold for this transition. What we did is, for different values of persistent current, we compute the minimal external input, which is necessary to initiate CSD. And we said that there is CSD initiation when there is a depolarization block in the pyramidal neuron. And we see that persistent sodium current reduces this threshold. And moreover, if we use the same external input, then CSD is initiated earlier when we have more persistent current. And this is consistent with experimental results. So those are results from our collaborators. They are using this toxin, which I mentioned, HM1A, to mimic the effect of the migraine mutations of NAV 1.1. So when there is this toxin, there is persistent sodium current. And they take brain slices of mice. And when the slices are bathed with the toxin, they can observe CSD in the neocortex, but they never observe it in the control condition where there is not the toxin. And if they use optogenetics to trigger CSD, then the CSD is initiated earlier with the toxin than in the control condition. And another group is using a mouse model for those migraine mutations of NAV 1.1. And they see that in the mutant animals, the threshold for CSD initiation is reduced, and the CSD latency is also reduced. So there are two effects. There is the reduction of the threshold and reduction of the latency, and our model reproduces both of them. Now I'm showing some voltage traces. The first row here, it's without persistent current, so it's the control condition, and you can see the two neurons firing. There is a little delay before the pyramidal neuron starts to spike. This is because there is an inhibitory synapse from the GABAergic neuron to the pyramidal one in our model. And here, there is some persistent current, so it's the migraine condition, and the two neurons enter depolarization block here and here. And now I will show some experimental traces where CSD is triggered using optogenetics. And if we zoom, we can see that the dynamics is qualitatively similar. So now for the simulations of the previous slide, I show the firing frequency of the two neurons, extracellular potassium and extracellular sodium. So at first, during the initial phase, the pyramidal neuron does not spike, just the GABAergic neuron is spiking. And potassium rises faster when there is persistent current than in the control condition. even though the firing frequency of the GABAergic neuron is slightly smaller. This is not intuitive, but it's consistent with what we found studying the effect of the mutation on the GABAergic neuron. So at some point, the pyramidal neuron starts to spike and the frequency increases a lot, and the frequency of the GABAergic neuron also increases until they enter the polarization block, so they stop to spike. And we think that this initial increase of extracellular potassium is crucial for CSD initiation. One day during a talk, someone asked me why we say it's the potassium which is important and not the sodium, because the sodium is also really affected by the mutation. So to answer this question, we tried numerical experiments. We tried to use very high diffusion rates for either extracellular potassium or extracellular sodium. And the idea is to keep their concentration almost constant. And if we keep extracellular potassium constant, this case here, for all the values of persistent current and external inputs which we tried, we do not observe CSD. So in our model, keeping extracellular potassium constant enables to prevent CSD initiation. And this is not the case when we keep the sodium constant. In fact, when we keep sodium constant, the threshold and the latency are even reduced. So from this, we are sure that it's the potassium which is the important ion for CSD initiation. And we tried the opposite. We tried to have smaller diffusion rates for extracellular potassium. And in this case, as expected, the threshold and the latency for CSD initiation are reduced. Okay, now I will move to the epilepsy mutations of NAV 1.1. So to model them, we reduce the conductance of the transient sodium currents. And as before, we first focus on the effect on the GABAergic neuron itself when it's not connected to the pyramidal neuron. We compute some input-output relationships. And so the number of action potential which are generated increases when we increase the input, until at some point there is a depolarization block of the neuron because the input is too strong. And this will be more clear in the next slide where I will show some traces. And this happens for smaller external inputs in the epilepsy condition. And the same is observed experimentally. Here they used a mouse model for the epilepsy mutation. So those are the voltage traces. On top is simulations. This is without the mutation, the neuron is firing. And when we have the epilepsy mutation, the neuron enters the polarization block, which is what causes this drop in the number of action potential. And experimentally, the same thing is happening. What is the consequence on the firing frequency of the pyramidal neuron? So here on top, it's the control condition. And here we have the epilepsy mutation. And when the GABAergic neuron enters the polarization block, it stops to generate action potentials. And this means that the inhibitory effect it has through the inhibitory synapse is removed. And at this exact moment, we observe a sudden increase of the firing frequency of the pyramidal neuron. So this is not a seizure, but we have here a model with only 2 neurons. So probably to study seizures, we would need network effects. Okay, so to conclude, we study within the same model epilepsy and migraine mutations of the NAV 1.1 channel. And to study one or the other of those mutations, we just modified 2 parameters which are directly related to the effects of the mutation, and otherwise the framework is exactly the same. And in both cases, we noticed that there are other mechanisms which are relevant, which are not modifications of the firing frequency of the GABAergic neuron. And this is interesting because experimentally concerning pathologies of neuronal excitability, the firing frequency is the main feature which is investigated. But here other things are important. Concerning the migraine mutations, in our model, they increase the ion fluxes at each action potential, which causes an accumulation of extracellular potassium, which facilitates CSD initiation through reduction of the threshold and reduction of the latency. In the case of the epilepsy mutations, in our model, they make GABAergic neurons more susceptible to depolarization block, and this is also something which is observed experimentally. And this is favorable for epileptic seizures because the inhibitory restraint of the GABAergic neurons is not there anymore. So to see if these are hypotheses from our model, which should be tested experimentally to see if they make any sense. So we hope to motivate some experiments. And what we are working on now is on developing a simpler model, simpler version of this model, because this model is quite heavy because it has a lot of variables, parameters, and we suspect that there are several timescales. So we are right now building a smaller version with which we would like to study the bifurcation scenarios for the transition to depolarization block. Okay, thanks for your attention. Those are the papers for this work, this one for the modeling aspects and this one for the experimental aspects.
Thanks, Louisiana. So we have plenty of time this time for questions. Thanks for finishing on time. So again, questions can go in the chat, hands can be raised, or as a backup, you can just unmute and speak up. I'm gonna pause for a minute and I'll get the ball rolling again. So yeah, you know, this point about whether it's the potassium or sodium that's responsible for the you know, depolarization block effect that you see is very interesting. And your numerical simulations show the dominant impact of potassium. Can you comment from a more like biological or just sort of intuitive perspective about why you think, you know, it's the sodium actually is not enough to, you know, push you into depolarization block in the same way as potassium?
I think it's because if the extracellular potassium increases, it will increase the reversal potential for the potassium, for the pyramidal neuron. But if the extracellular sodium decreases, it has the opposite effect on the reversal potential. So if the reversal potential of the potassium increases, this is like a depolarizing effect, which can promote CSD initiation. So
right, but so the sodium reversal you're saying becomes lower, so it's sort of less depolarizing.
If we decrease the extracellular sodium concentration, this reduces the extracellular the sodium reversal potential.
Right.
But for the potassium, it increases it. So.
Right. But so with this lowered potential, in theory, then it becomes, you know, your spikes aren't as large.
Yes, it's true. But this is why then we tried to keep either one or the other constant, and we found that keeping the extracellular potassium constant prevents CSD, but not for sodium, and in fact the opposite.
Yeah.
Can I say something? Excuse me. Hello.
Sure. Yeah, go ahead, Gare. Yep.
Do you hear me? Yeah. Yeah, I think Louisiana is right, and also, but also I think that isn't it so that in general the relative change is actually larger in potassium because you have a very low concentration.
Yeah, this is true.
So that it actually changes more than the change in the reversal potential for potassium is larger than for sodium, the relative change.
Yeah, because extracellular sodium is very large, and extracellular potassium is like around 5 millimolar.
Yeah.
Sometimes when you change the reversal for sodium, you can have this sort of counterintuitive effect whereby pushing it lower, because you have the smaller spike, then you also affect how much potassium is coming in or out of the cell. So it can get non-intuitive, but I agree with your point, Gare. The magnitude of the change could be very important.
Yeah, but you have actually checked it, so I guess it's like, yeah.
We have another question in the chat from Shuiji Li. Sorry, I'm just going to go ahead and read the question. So I suppose depolarization block only happens in the conductance-based neuron. Correct me if this is wrong. If so, does the model depend on the neuron model? For example, if you had leaky integrate and fire neurons, then you would not get such effects. Is that correct?
I don't know, because I don't know about this thing that it does not happen with the other model. So I cannot answer the question. I just know that we tried it with conductance-based models.
Right. I mean, I guess it's clear that if you don't, you need somewhere for the ion concentrations to show up in the voltage.
Yes.
That would be a minimum requirement. Yeah. Okay. So we still have time if there are any additional questions.
Maybe I can ask something.
Sure.
You talked about diffusion in your model at some point.
Mm-hmm.
Was that implemented just as something that disappears from the system?
Okay, I can share my screen to show the equation. No, I did not put it there. This is simply like we have a rate, and then we have the difference between some bath concentration and the current extracellular potassium.
Yeah, right.
Which will then, it's this term here.
Okay.
Okay. And then another question, this one's from Hillmeyer. So is the sodium potassium pump in your model always strong enough to bring you back from depolarization block?
As far as I remember, we did not have cases where it does not happen.
Phil, do you want to unmute? Is there like a specific scenario you have in mind? No, he's not taking me up on it. Okay, go ahead.
In the case of the epilepsy mutation, we did a bit of bifurcation analysis. And when we have the epilepsy mutation, we have this bifurcation diagram here where we take extracellular sodium as a slow variable. And so once we are on this steady state here, we don't have a way to come back. But I don't know how this is related to the sodium potassium pump. And in the case where we have the control condition, we have, in fact, bursting. So we go to the polarization block, but then we spike again. And this bursting is a property of the model we use for the GABAergic neuron. It does not really answer the question, but I don't know more.
If you change the pump strength, do you push towards the upper scenario or something, if you increase the pump strength, for example?
We did not try it.
Okay, great. Well, thank you, Louisiane.
Thanks.
So Gary, you can go ahead and share your screen.
Thanks. Do you see this?
Yeah, you're showing up, but you're in the mode where we also see your notes.
Really?
The sort of presenter mode.
Am I still in that mode? Okay.
Right now we don't see your slides.
Okay.
We still have a minute.
What about no?
This is good. Yeah,
This
that's
is good.
perfect. Yeah.
All right. So no, I can't see my notes, but I don't have so many notes, so I think I can do without.
Okay, that's Wait 1 minute and then we'll get going. Just to keep on track exactly.
Yeah.
At least according to my clock.
So I can say thanks for inviting me. That doesn't have to be a part of the formal talk, but thanks anyway.
So yeah, I'll just introduce you. So Geir Helms from Norwegian University of Life Sciences is our final speaker of the session on ion concentration dynamics in brain tissues. So Geir, the floor is yours.
Thank you. So it's kind of a broad title, I suppose. So it's effects on neurodynamics and extracellular potentials, and the two are kind of different when it comes to modeling formalisms needed. And I'm in this group of a guy called Gaute Einevold. And the main expertise in this group is on modeling extracellular potentials. And so I'm going to start a bit just putting up the standard framework used for that. So when you want to model it, you typically model the neuron using some kind of multi-compartment model, for example, based on the neuron simulator. And then you compute the neurodynamics using that. You get currents going in and out of the membrane. And then when you're done simulating the neurons, you can, in step 2, feed these transmembrane currents into the surrounding tissue as just, you forget about the neurons and now you just have a set of distributed sources and sinks. And if you have such a distribution of sources and sinks, you can actually find an analytical solution for the extracellular potential that you will have outside this neural activity. And this equation is based essentially on current conservation, so that you will have closed loops between all these sources and sinks. And just to compare it to the previous talks that have been given, I think all of them were essentially single compartment neurons. And which means that you only have an inside and an outside, and you have no space inside the neuron. And in that case, you would not have any extracellular potential, because you would have to have, because of current conservation, you will have to have the same current going in to the neuron in form, for example, of a sodium current, as going out from the same point in the form of a capacitive current charging up the membrane. So you would have a zero sum. Not zero voltage dynamics, but zero current, and then no extracellular potential. So the extracellular potential depends on the distribution, so that in this case, you can have current coming into the soma, going up the axon or dendrite, and out somewhere else. But you do this using a two-step scheme. And the main assumption in such a two-step scheme is first that there's no feedback from whatever the neurons causes the extracellular potential to do, doesn't act back on the neurons, because you have simulated the neurons in an independent step. And normally you assume this is okay, because the extracellular potential is so small compared to the membrane potential that it wouldn't affect much anyway. That's the assumption. And the second assumption is that ion concentrations are constant, as you have talked about today, and which is also the main topic of my talk. And a third assumption is that this extracellular current is ohmic. It's given by Ohm's law. And this is a prerequisite for deriving this equation that you see down here. And based on the previous talks, I also think that you know this fairly well, but I'll repeat it quickly. So the reason why we sometimes can assume constant concentration is that if you look at the concentrations inside the neurons, they pretty much sum up to a zero net charge. So the number of anions and cations are more or less the same. And so there's a small deviance here, 0.2. Maybe that's an overestimate, but it still is like on a decimal place. And this net charge inside the neuron, it actually not floating around in the bulk solution because that's electroneutral. It's sitting on the membrane, and it gives rise to the membrane potential, which for resting neuron is something like minus 60 millivolts. So what happens during an action potential is that some sodium rushes in. It changes, for example, the sodium concentration by 0.3 millimolars. And then it repolarizes, and then some potassium goes out and changes the potassium concentration, reduces it inside. And the assumption is that between this action potential and the next, the pumps have time to pump back these original, reset the original concentrations. And also the assumption is that these tiny concentration changes during an action potential are so small that you don't really have to bother with them. They don't really change anything in terms of neural dynamics. And all this, of course, fails in some cases where you have hyperactive neurons that are firing too fast for the pumps to keep up, or if the pumps in themselves are not working, such as after a heart attack or a stroke or decapitation. So when are these concentration effects relevant? Of course, they are very relevant in very tiny spatial scales, such as inside spines, ion channels, synaptic clefts, and very close to membranes and the bilayers. And this is not my focus. I focused on the larger spatial scale, and that's when neurons fire fast over time, and you get an accumulation, for example, of potassium in the extracellular space. Potassium seems to be the most critical, as I agree with the previous talk on this. And then you get changes in reversal potentials. And also, if you really want to do this spatially, you get changes also in currents going through extracellular space. They are not ohmic anymore, they are also diffusive, meaning that there will be currents proportional to concentration gradients because ions are charged, and when charges move, it will be an electric current. And so part of my title was extracellular potentials, and I've devoted only this one slide to it, and I focus later more on the neural dynamics aspect. So we've focused in quite, well, 3 papers on how diffusion can affect extracellular potentials. And we've kind of simulated, and I'm not going to present the formalism behind this, but we simulated it in this way. We took a complex neural model, but standard in terms of implementation. It's a neuron simulation in a model from Hay and co-workers. And we ran it for 80 seconds simulated time, and we computed the currents going over the membrane at all different neural compartments. And we split it into sodium, potassium, calcium, and unspecified and capacitive currents, and we kept track of all the ions going out into this box of extracellular space. And then we looked at the concentration due to electrodiffusion in this extracellular space. You see this red blob surrounding the neuron. This is the change in potassium after 80 seconds of simulation. And this panel below shows for a specific location during an action potential, the extracellular potential in one case where we accounted for the diffusive effect on it, and in one case where we didn't. So this orange line would be the standard theory, and the blue would be if you account for diffusion. And what you see is that they're essentially parallel, but diffusion has only shifted it by like a small amount to a more negative value in this case. And so this shift, you could picture that in the beginning of the simulation, where potassium was more or less constant in the entire space, these would be on top of each other. And then during 80 seconds of activity, changing the potassium concentration, the concentration has shifted the potential by this much. So it's a very slow effect. But if you look at the power spectra for this with and without diffusion, you can actually see that for high powers, the 2 cases are on top of each other, but for lower power, you see they split apart, so that you could expect diffusion in some cases to show up in your power spectrum if you're measuring local field potentials and things like that. So that was my first part of the talk. I called it an introduction, and I put all this extracellular stuff in the introduction. And now I go on to a kind of general mathematical framework for dealing with these kind of things. So you can think of this problem as having three different kinds of currents: those that go across the membrane, those that go inside neurons, and those that go outside. And the membrane currents, you know well, Hodgkin-Huxley type, and the only ghost of ion concentration is baked into this reversal potential, and you normally assume it's constant in standard models. And intracellular, you have the Cable equation, voltage gradient, the currents follow that, and the same in the extracellular. The difference is that you have 3D. So what happens when concentrations start changing is that, as you have seen in the previous talk, reversal potentials can change, and that will change firing properties. And so you get this depolarization block and stuff like that you can simulate. And if instead you get gradients in the extracellular space, I focus on the outside here, you cannot use Ohm's law anymore to study it. You have to use the Nernst-Planck equation for electrodiffusion, where you have a flux of ion going because of the concentration gradient, CK, and another one because of the potential gradient, because if there's a potential gradient, it will propel ions along with it. And that should replace Ohm's law from the standard models. And just to look at the relationship, I can sum over all fluxes weighted with their valences to get a diffusive current and a field-driven current. And then I can compare this to Ohm's law, and it should be the same if this diffusive component is 0, and if this prefactor, concentration-dependent prefactor, is equal to sigma, the conductivity, and if that's constant. And so when we want to solve this, we have to solve this equation. And if we say, maybe ideally, we would like to just demand that this should be fulfilled everywhere in space. And we have two options, because this will give you one equation for each ion species, but we need one additional equation for this additional variable phi, which is the voltage. And we have two options. One is to use Poisson's equation, which derives phi from the charge density locally. And the problem with this is that it's extremely computationally efficient, because this charge density accumulates on a nanosecond, nanometer scale, and you have to have an extremely fine resolution. And there's an easier way, and that is to just assume that all solutions are electroneutral, and so that the charge density should sum up to 0 everywhere. And to explain, I'll jump to the next slide. To explain the difference between the two, I can try to explain what the diffusion potential is. So if you picture that you have a box with two ionic solutions of different concentration, consisting of anions and cations of the same amount on each side, and you put them in contact with each other at t equals 0, time 0, what you will expect is both to start diffusing towards the right, towards the lower concentration. But if you look up in tables, you will see that, for instance, if this is sodium chloride, you will see that chloride has a larger diffusion constant than sodium, so that due to diffusion, more chloride will move than sodium. And this is a quite dramatic thing, because now you have a net charge transfer from one side to the other, and it will give you a charge difference between the right and left box. And this charge difference will set up a potential, almost a step potential between the two sides. And this step potential will slow down the chloride ions and speed up the sodium, because they're oppositely charged, so that you get a 1-to-1. And this you will get after just about 10 nanoseconds or so. So that difference between these two frameworks is that the Poisson-Nernst-Planck, you actually have to simulate this very fine-grained charge relaxation process, while in this electroneutral scheme, you just calculate the step potential needed for it to become one-to-one. So it's a shortcut, and it can be shown to be accurate on timescales larger than 10 nanoseconds, and at least on the micro millisecond timescales that you are interested in when simulating large-scale neurodynamics. So the essence of this framework is essentially just to solve the Nernst-Planck equation in the intra and extracellular space, but you don't want to solve it in the membrane, because that's like a very complex place with the bilayers and everything. So instead, you couple them on the membrane using Hodgkin-Huxley type boundary conditions, and you can compute the potential by demanding these two, by putting in these two constraints, that for any compartment not bordering to a membrane, there should be no net charge at all times, and for any compartment bordering a membrane, the net charge should be exclusively associated as a membrane charge, and the membrane charge will give you a membrane potential. So there should be a strict relationship between the charge in the in and outside and the membrane potential. And so maybe that's, from a technical point of view, that's the model we are most proud of now, because we have a way to do this for a general 3D geometry. But the problem is so you can see here we have meshed up both the extracellular and intracellular space, and we simulate ion concentration dynamics in both compartments and make sure that the membrane currents are consistent with that. And the problem with it is that it's extremely computationally expensive. So if we want to run simulations on it, we are limited to just very small idealized geometries so far, and maybe brief simulation, maybe some hundreds of milliseconds, and a few pieces of axons or something like that. And so in a sense, I think it's a bit boring to play around with models like this, because you have to start to simulate. We have to do a lot of work to set up your simulation, and you have to trust very much the parameter choices that you made, and then you have to wait for a couple of days, and then it spits out a result. So it's more fun to do a smaller model where you can just run a lot of simulations and play around and see what happens. So I'm going to focus this last short part of my talk on just commenting on a minimal model that has it all. So we wanted to see if we can make a full multi-compartment model which is completely electrodiffusive in its nature, meaning that it accounts for ion concentration dynamics, spatial propagation, and has a consistent relationship between all ion concentrations and voltages everywhere. And by minimal, I mean that multi means two in this case, and that we have different ion channels in the soma and dendrites, so it has some spatial structure and can get somatodendritic signaling. And also we have a spatial explicit extracellular space, but also here only two compartments. And that this model should account for all ionic movements due to electric drift, diffusion, and transmembrane currents. And so the way that we proceeded trying to make this model was to take an existing two-compartment model. So we used this Pinsky-Rinzel model, which is fairly well established. It has two compartments, a soma and a dendrite, some potassium channels and calcium channels in the dendrite, the standard action potential channels in the soma. And then we asked, can we make this ion concentration, can we combine this with ion concentration dynamics in a way that makes it provide the same functionality in terms of firing pattern as the original one, which means that we cannot assume that ion concentrations will remain constant, but we actually have to make sure that they do. And I wanted to comment on something before I go on, and that when we equipped this with an intra and extracellular space, we assume that the extracellular space has half the volume of the intracellular, which is a tissue average. Neurons occupy about 40% of brain tissue, glial cell 40%, and the extracellular space 20%, so that extracellular is small. And when we do this with closed boundary, as I also think that to some degree other single compartment models have done, we actually assume periodic boundary conditions, meaning that when my neuron pumps out some potassium and it ends up and stays in this compartment, why doesn't it diffuse away from here and use the whole space? Well, in this sense, it wouldn't if the neighbor was doing exactly the same, because then you would get the same concentrations everywhere. So this is, it should be noted that that will represent the scenario where you have perfect synchrony in a population. It was just a comment. Apart from that, I think this model is nice, but this gives you this particular interpretation of what it should mean. And so just some few take-home messages from this model. What I found interesting was first to see how long time it actually took to get a resting state. As you maybe know, if you have made these standard models, normally if you don't know exactly what the resting potential in your complex model will be, but you just run your simulation and let it settle on some resting potential. If you're lucky, it has one. And that normally in a normal model, it takes a few hundred milliseconds perhaps. So you let it just run for 100 milliseconds before you start doing something with it. But in this case, when you have ion concentrations that can go back and forth and even inside and outside, you have a lot of different variables that need to find their collaborative resting state, and these concentrations are very slow. So what I've found in this model, what we found was that we have to run it for more than 1,000 seconds of simulated time before it actually gets entirely to rest. And it doesn't even change much during these 1,000. It changes by a few millimolar and finds its own resting state. But it has a resting state, and just to show, or like one criterion for this to be an acceptable model was that it should reproduce the spike shapes from the original model. And it did. It's not very surprising since it contained the same ion channels. But so now we have a more complicated model that does exactly the same, which is normally nothing that you applaud. But at least in this case, you can compute a set of new variables that you couldn't in the previous model, like so concentration shifts during spiking and conductivity shifts in the extracellular and intracellular space, energy consumption due to how much the pump did pump, and the extracellular potential. But the main point was that, and this was something that we hoped would happen and expected to happen if the model was sound, that if we stimulate this with a weak input so that it's firing slowly, it changes during firing the potassium concentration, but it settles on some dynamic steady state, and it can go on forever spiking, and it can spike identically to the original Pinsky-Rins model. But if we stimulate it harder, the concentrations will drift off gradually, so it will spike identically to the original model in the beginning of the simulation, but after a while the concentration changes started to show up in the spiking patterns, and in the end it reaches the polarization block. And I think I saw something like this in David and Luzan's talk too. And since I am running out of time, I'm jumping over this. I wanted to just make my last comment on the tendency in the field of decoupling electrical drift and diffusion, because standard models of electrodynamics normally you only consider drift currents, which means only the ones that are driven by the potential gradient. While if you want to model concentration dynamics, you often do the opposite. You only consider concentrations changing due to diffusion. So, and we asked during, or the motivation that for looking into this was that I spoke to a colleague who wanted to study some sodium-dependent stuff going on in dendrites, and he wanted to model diffusion of sodium from soma towards dendrites. And I wondered, is that a meaningful thing to do? Because there are all these electric currents going in that direction, and they are also carried by ions that are there, and maybe sodium moves just as much due to drift as diffusion. So we asked, during this particular simulation that you see here, what causes ions to move from soma to dendrite, drift or diffusion? And the result is shown here. Blue is drift and orange is diffusion, and the panels show a cumulative movement of charge or ions. And I think this colleague was right. In the case of sodium, it was mainly diffusion that drove sodium upwards. But in the case of potassium and chloride, which have higher concentrations on the inside, by the way, you can see that at least drift and diffusion are of comparable magnitude, and so on. So I cannot conclude that these two phenomena can generally be decoupled as you would like to do. We have expanded this by adding a glial cell outside, and I'm not going to talk about that. It's available in a preprint. And thank you.
Thanks a lot, Geir.
Sorry for going over time.
We do have time for questions, and we have one in the chat. So this one comes from Mark V. What are the exact conditions for using KNP versus PNP? It must depend on the length scale as well, presumably.
Yes, that's like the troublesome question. But I think if you use PNP, you need to model the actual accumulation of charge in a Debye layer. So typically, if you do that around a membrane, that will be like 10 nanometers or something like that at max. So you have a concentration gradient. So you need a spatial resolution of smaller than nanometers and a temporal resolution smaller than nanoseconds. And if you don't use that, your system you will get all these instabilities, ringing and stuff like that. So it's very impractical, but it's very accurate. KNP, you can just choose any sort of temporal and spatial scale. It will not be as accurate, but it will be stable, however you choose it. I hope that was a somewhat satisfactory answer. KNP, you cannot study phenomena taking place just immediately outside membranes and inside ion channels and at a very fine spatial resolution. But like on a tissue average or whole cell level, I think it will be fine.
Okay. Another question is this 1,000 second requirement for reaching steady state is quite striking. That's quite a long time. Is this because your standards for being converged are really, really strict, or is there can you comment on any biological effect that requires this much time?
Do you still see my screen?
Yes.
Okay, so I'll just, I've lost it myself. Okay, so it's not really strict, but anyway, you can see the convergence here somehow after the concentrations have changed due to a period of spiking. So you can see they start leveling off towards rest. And in this case, the time scale is 6, so it's a minute. So in this case, it takes about a minute to reach. And I think our criterion for convergence was something like reached delta concentration should be less than 1% or something like that from something you get after 5,000 seconds.
Okay, so it sounds like this 1,000 seconds is not an absolute number, but it's more a function of what type of transient and stimulation precede the convergence.
Indeed, yes. But in general, I mean, the point was just that it's like on a minute time scale, I think you could say, where you can expect concentration effects to linger in your system for a minute before. So it has a memory. Concentrations gives this cell a memory of a minute, in a way.
Okay. We've reached the end of our time. I don't know if there's any other quick question. Okay, yeah, well, it is noon here, or 6 p.m. I guess in continental Europe. So that reaches the end of the session. So thanks very much to David, Ryan, Louisiane, and Hare for these really nice talks, which have been recorded and therefore I guess will be available for us all to recommend to our colleagues and so on once they're processed, by Daniele and his team. So thanks again, everyone, for participating, and I really enjoyed the session and hope you did as well.
Well,
Yes.
Thank you very much, Jonathan, for organizing the session and for all the speakers. I should just say that, as you know, tomorrow there will be a panel discussion in which we're going to discuss a little bit the future of ICMNS, and you're welcome to already contribute with questions or themes. There is a link to the book in the booklets that we sent you the other day, and yeah, so if you want, just contribute to that. I will now terminate the recording and the YouTube stream. Thank you, everyone.
All right. Thanks, Daniele. Bye-bye.
Thank you.